Hello everyone, this is Dr. Khurram, your IGCSEs and A-levels biology instructor. And for today, we will begin discussing the topical past paper question for one of the technical topic that you have for your IGCSEs 0610 biology course, and this course with the name of plant nutrition. And simply, like in this chapter, you may have come across the major metabolic process that we have caught occurring in the plants, and this goes with the name of photosynthesis, we say. So if we just look on toward this question that appeared in February, March of 2015, and this is coming from the structure of the leaf. And what the examiner has to tell you is that we give you figure 1.1, and this is a diagram of a cross section of a dicotyledonous leaf as seen using a light microscope. Say they took a cross section from a dicotyledonous leaf, and now they are showing you what they actually came across in the microscope. Now, if you were to see clearly, at the top here we have got the cuticle. Immediately below the cuticle we have got these cells of the upper epidermis. Then here we have the cells of the palisade mesophyll cells we have here, like each individual cell is palisade mesophyll cell. Then here we have got the spongy mesophyll cells. Here we have the air spaces. If I come downward at this point, so these are all the lower epidermis cells. Here we have these guard cells, like two guard cells are shown here, this and this. And in between them, we have this opening, and this opening, is stomata with us. So now, if we just move to the questions, they say is that you are required to be naming the tissue A and the cell B. So if the examiner is saying that you have to be naming this as a tissue, so you would never call them as palisade mesophyll cells because that would make it a cell. But since we have to regard to the tissue, so we would say this is palisade mesophyll layer. So that would relate to the tissue. And if they have asked for us to be naming the cells that we have at B, so if I were to just zoom in more, so this cell is the guard cell with us. Moving onward, he asks us that describe two ways in which the tissue A is adapted for the maximum photosynthesis. So you see the tissue A was the palisade layer, and it has a lot of palisade mesophyll cells in it. And what we know is that these palisade mesophyll cells, if you just look on their diagrams, so they have got abundant or many number of the chloroplasts in them, and that means that they would absorb maximum of the sunlight for the photosynthetic process. So if I just take you to the answer, so since we just have to describe it, so we would say that they contain many chloroplasts. And the other scenario is that these palisade mesophyll cells, they are positioned near to the top of the leaf, and that would help them to be absorbing maximum sunlight. But in terms of description, you would only say that they are present near to the top of the leaf. Another alternative point could be that in these palisade mesophyll cells or the palisade mesophyll layer that we have here, the chloroplasts, they are aligned near to the peripheries, like they are projected outwards, so this increases their exposure to be getting a lot of sunlight. Moving onward, they ask us that the plant, they use carbon dioxide for photosynthesis, and we are required to describe where and how carbon dioxide enters a leaf, like where in a plant this enters and how this would enter. So what we know is that on the lower side of the leaf, what we have with us, that is the lower epidermis. And in between the lower epidermis, what we have are these bean-shaped kind of the cells. And these are the guard cells, isn't it? And if I were to just simply tell you, in the external environment, we have the carbon dioxide gas, but comparative to the air spaces that we would have inside, the concentration of the CO2 here would be higher. So where this enters into a leaf, so we would say either we would say in between the guard cells or through the stomatas. And then how this is entering, so this would simply diffuse from being here in the higher concentration to the lower concentration that we have in the air spaces that exist within the spongy mesophyll cells. So since the marks were three, so what we would say that the carbon dioxide enters the leaf through the guard cells, and it would diffuse in context of how. So it would diffuse from an area of the higher concentration that lies here in the external environment to the lower concentration that lies within the air spaces of the spongy mesophyll cells. Now for 1 mark, they have asked you to state the two products of photosynthesis. So what we know is that when the plants they are photosynthesizing, so they would be combining 6 moles or 6 molecules of the carbon dioxide gas with 6 molecules of the water. And as a result of that, we would end up making a glucose molecule out of it. And the plant chloroplast they would be releasing 6 molecules of oxygen gas. So the two products of the photosynthesis, since we have to state them, so one of them is glucose, and the other one that we have with us, so this is the oxygen gas. Now, if we come here, this is coming from the May June of 2015, and in your exam, this was question number 2 with us. And in this question, we will try to see that how come the question can come with respect to an investigation or where the examiner would try to see your approach in terms of limiting factor of photosynthesis. Now, what they have to tell us is that there are students and they are carrying an investigation where they are interested to find out that how come the carbon dioxide concentration would impact the rate of photosynthesis and that too in an aquatic plant. Now, since in this experiment the students they are looking on toward the effect of the carbon dioxide concentration, so they would change it. So this would be acting as a variable, and that variable it would go with the name of independent variable. Like I would be repeating the investigation, and every time when I'm repeating it, we would be having a different concentration of the carbon dioxide gas, and by that we will see that how the rate of the photosynthesis is impacted in the aquatic plant. Now they have given you the diagram below, and over there they have shown you the apparatus that was used by the students. And when the students they carried out the investigation, so they were advised that the light meter, like they did not show you the light meter here, but they would for sure keep some light meter here, and that would be positioned at the same distance from the lamp. Like if we have got this movable lamp that we can either move it toward the plant or away from the plant. So the light meter, they would have placed it at the same distance from the lamp. Like if I am placing the lamp here, so the light meter would be placed here as a pond plant. And now they say is that the student counted the number of the bubbles gas produced and by the cut end of a stem. So we are measuring the bubbles of the oxygen gas, and since this is something that we would be saying that this is dependent variable, like you should remember this for your ATP and your P4, that anything that we measure in an investigation, so this would go with the name of the dependent variable. Now the scenario that you have to consider here is that they have taken a test tube, and in the test tube they have added this aquatic plant, and then they have also included the thermometer. Now, why they are including a thermometer here so that they can monitor the temperature of this test tube in which they have immersed aquatic plant. Because by some mean or the other, if the temperature would fluctuate, then we would not be able to see the impact of a change in the carbon dioxide concentration, which was the motive of the investigation of the student. So this temperature thermometer would give us an idea that what temperature is there in this test tube. At the same time, now you see that they have immersed this test tube in the water bath. Now, here we must be very much clear that why this water bath is being used, because you see that this lamp it would be emitting the light rays, and these light rays, they always carry the heat along with it. But since I'm investigating the impact of the CO2 or the carbon dioxide concentration on the rate of photosynthesis, so at any cost the excessive heat that would be emitted by this lamp, it should be absorbed. So that is why they have placed this test tube into this water bath. And now they have also placed this ruler here. And why this ruler is being placed? So that the distance of this lamp, it should be kept constant. And when we would keep the distance of this lamp at a fixed position from this aquatic plant in the test tube, so by that our light intensity, it would remain constant. And at the same time, they have added this dilute sodium hydrogen carbonate solution here. And you must know that this dilute sodium hydrogen carbonate solution, this acts as the source of the carbon dioxide that this aquatic plant would be using for the process of photosynthesis. And by that, when the photosynthesis would be accomplished, so we see the bubbles of the oxygen gas they are being released. And this is something they are measuring it up. So more the bubbles of the oxygen gas it would be proportional to more of the photosynthetic process that is occurring. Now, if we were to move to the questions, they say is that explain why the students included the following in the apparatus. And for 2 marks, they are asking the beaker of the water and the thermometer. And as I have told you above, that the beaker of the water that we are including, so this is used to absorb the heat which is emitted from this lamp, because temperature is something that we have to be keeping it standardized, or we need to be keeping it as a controlled variable. And the thermometer which they have included or they have added into the test tube at this point, so this is actually being involved so that we should get a track of the temperature, that what temperature lies into the test tube in which the aquatic plant was immersed. If you forget any of the points, alternatively you could say that the water beaker was included so that we should prevent any change in the temperature, because we are looking on toward the impact of the carbon dioxide gas concentration change on the rate of photosynthesis. Now moving onward, for another 2 marks, they are asking that why did they include the light meter and the ruler. So the light meter would be included with an intention to measure the light intensity, and the ruler would be used to keep the light intensity constant. Like again, you see that I have to be keeping the light intensity, I have to be keeping the temperature constant, because our investigation motive is to be looking the impact of the carbon dioxide gas concentration on the rate of photosynthesis. So if your answer would look like this, so you would score 2 marks for this. Now moving onward, here there are some of the technical points that you need to have an idea of, and they tell us is that the results of the investigation which were obtained by the students, they have shown you in this figure 2.2. And from this graph, we are required to be understanding that at what point something becomes limiting and something becomes excess. So if I were to write it down, there are a total of three of the factors that can impact the rate of photosynthesis. One is the carbon dioxide gas concentration. The other thing that we have with us, so this is the light intensity, because the light intensity is something that would interfere with the rate of photosynthesis. And the third thing would be the temperature with us. Now you see is that this CO2, since we were changing, so this is the independent variable. And the light intensity and the temperature, they would be the controlled variable. So this means that from the very beginning of the investigation, the light intensity and the temperature, they would be kept controlled, but the CO2 concentration, it would increase from 0% to a higher percentage. So if we were just go on toward the x-axis, on the x-axis they have given you the carbon dioxide gas concentration in terms of percentage. And if you were to look clearly, so this is it being increased, its concentration is being increased from 0%, and max this is coming till 0.50. And if we just look on toward the y-axis, here they have given you the rate of the photosynthesis. So this is our dependent variable, and the rate of photosynthesis is proportional to the number of the bubbles that would be produced per minute. What you have to be remembering is that any of the thing that comes here on toward the x-axis, which is the carbon dioxide concentration. So in this part of the graph, this would act as a limiting, and the other two factors, which is the light intensity and the temperature, they would be excess. But if we just look at this point where the graph is becoming constant, or the graph is leveling off, or this is forming a plateau, so from this point onwards, anything that is given here on the x-axis, that would become excess, and the other two, which is the light intensity and the temperature, they would become limiting. Now, if we were to move for 3 marks, it says that you are required to describe the student result, and you would gain the credit if you would use the data from figure 2.2. Like the description is something very easy. Anything that is as shown in the graph, you are required to just simply say it or write it up in terms of number values. The first point should be general, and the other 2 points in context of 3 marks, they should be very much specific. So if I were to describe the graph, so what I see is that as the carbon dioxide concentration increases, the rate of the photosynthesis, this increases, and then this levels off. So this is one general point for description. The other point, if you just see, is that initially when the carbon dioxide concentration is being increased from 0.00 to 0.10%, so the rate of the photosynthesis, this is increasing but at a slower rate, and we were only able to produce 5 bubbles of the oxygen gas. Okay, so this is a specific description point with context to these values and this value. if I were to tell you another description point, so you would say that when the carbon dioxide concentration is being increased from 0.10% to 0.40%, so the rate of the photosynthesis, this increases steeply from producing five bubbles to ultimately producing the maximum bubbles of the oxygen gas, and that would be the 20 bubbles with us. So this is again another point for the description. Like the important point is that anything that you quote your examiner from here, you have to relate it to here as well. The other thing would be, other description point would be that at the carbon dioxide concentration of 0.40%, the maximum rate of the photosynthesis is attained, and for that we have produced 20 bubbles of the oxygen gas. Moving onward, another description point we see here is that when the carbon dioxide concentration is increased beyond 0.40% and going till 0.50%, so the rate of the photosynthesis, it remains constant, it does not increase further. So here I have said multiple description point. You will have to write any three with respect to the marks. So I am just reading it out for you that as the carbon dioxide concentration increases, the bubbles of the oxygen gas release they increases, as I've told you above, and then it levels off. This is one general point. And then I have written that the increase in the rate of the photosynthesis is slow up to 0.10%, where we have just produced five bubbles per minute. Okay, because they have given in term of minute. And then I have written the third point that the rate of the photosynthesis become constant or it levels off at 0.40% concentration of the carbon dioxide, like this point I am referring to. And here we were just producing 20 bubbles of the oxygen gas per minute. Now here for 1 mark, they have to be asking us that state the factor that is limiting the rate of the photosynthesis in the region X on the graph. Now if I just go back toward the curve, so here you see they have given you X here, and they are saying that what is limiting the rate of photosynthesis here. So as I have told you, that anything that comes on toward the x-axis, that in this part of the graph from here to here, this would be acting as a limiting, but where this levels off or form the plateaus. So this thing which is labeled here, so it would become excess here. So this is how you would score 1 mark here. And now for the total of 4 marks, they are asking that you are required to suggest and explain the reason for the shape of the graph in the region Y. Now, what you see is that we need to suggest and give the explanation that why come the shape of the graph appears as it is, like in the form of a plateau or leveling off at Y. Now again, you need to listen it very clearly that at Y, what I know is that this light intensity and the temperature that we have, they are becoming limiting. And the carbon dioxide gas, you see, from this point onward, it has got a maximum of the value, so this would become an excess reagent. And the light intensity and the temperature, they would become limiting. And why the rate of the photosynthesis is not increasing beyond this point, the reason is that when the light intensity is becoming limiting, so the rate of photosynthesis cannot be further increased because light intensity is something that would provide the energy for the photosynthetic process to proceed on. Moreover, the temperature you see again is becoming limiting here. And why come this would not allow the rate of photosynthesis to increase beyond this value? The reason is that the photosynthetic process is an enzyme controlled reaction. And when you have increased so much of the carbon dioxide gas concentration, but the temperature from the very beginning of the chemical equation or from the very beginning of the investigation was kept controlled, was kept constant, so it would allow the enzymes to be carrying on the photosynthetic process at that one specific rate. So if you just simply look on toward my answer that I have written from your mark schemes and the examine report, I have written that at Y, the rate of photosynthesis is becoming constant, and this is due to carbon dioxide that is becoming excess, but the light intensity and the temperature they become limiting. Why? Because they were kept constant from the very start of the investigation. Now we have to explain it. What I have written here is that light it is important for the photosynthesis because this provides energy for the photosynthetic process to proceed on. And as far as the temperature is to be bothered about, so this would influence the enzymatic activity, and the enzymes are what that basically regulate the photosynthesis. So when the temperature becomes limiting, the enzymatic reactions cannot be increased beyond that point where this actually become constant or it leveled off. Moving onward for 3 marks, they are asking us that counting the bubbles may not be the best way to measure the rate of photosynthesis. As I have mentioned above, that counting the bubbles of the oxygen gas is proportional to the rate of photosynthesis. And how they were counting, how they were like measuring this rate of photosynthesis and simply counting the bubbles of the oxygen gas that were released by the plant. They say the volume of the bubbles is not always exactly the same. The reason is that at some time the bubbles produced are more in diameter, at some point they are less in diameter. So this is not an accurate way or an efficient way of measuring the rate of the photosynthesis, and they want your suggestion and explanation where we are required to tell him one other alternative way of measuring the gas given off to solve this problem. So I would just simply indicate it with the help of the diagram. You see, they gave you a beaker at the top, and then there was this test tube with us, and here they have immersed the aquatic plant into it. And they said that the bubbles of the oxygen gas release, this is not an accurate way. So the alternative point could be that we need to be adding the rubber bungs to the top of this test tube. And then we would attach it with the help of the capillary tube to a gas syringe. And any of the volume of the oxygen gas that we would collect here in the gas range, we would divide it by the time, and that would be more accurate way. Another alternative way of collecting the gas of the oxygen in order to be calculating the rate of the photosynthesis would be that I would take a delivery tube from here, and that delivery tube, it would have the bubbles of the oxygen gas coming into it. Now, what we would do is that we would cause this oxygen gas to be displacing the water that we would have in an inverted measuring cylinder, and here we would have the water trough. Okay, so initially when the oxygen gas would not be coming into this inverted measuring cylinder, all of this inverted measuring cylinder, this would be filled up with the water. But as soon as the bubbles of oxygen gas, they would start to collect here, so they would push the volume of the water downwards. So any of the available space that we would create here by the collection of the oxygen gas, that would give us an idea of that how come or how much of the volume of the oxygen gas was collected in a given period of time. So how you were required to be writing it in terms of the points. I have written like two answers. So I have said that you are required to be adding the rubber bungs to the test tubes. And then we would attach the capillary tube and then we would connect it to the gas range. And any of the volume of the oxygen gas that we have collected, we would divide it by the unit of time. This is an alternative. The other point could be, as I've said it, we would pour the oxygen gas through the water trough and into an inverted measuring cylinder. And as the gas would collect, so the water would be pushed downward and we would have some markings here. And any of the space that would be created here in the inverted measuring cylinder, that would be proportional to the volume of the oxygen gas collected. Moving onward, this appeared to come in the year 2015, and this was question number 3 for your theory paper. And here they have said that the researchers in the Michigan, they investigated the rate of the photosynthesis in the leaves of big tooth aspen trees. So we have got these big tooth aspen trees, and we are removing the leaves out of them, and we are like simply investigating the rate of photosynthesis in them. And they have given you the scientific name of this aspen tree, and the genus is Populus, and the species name is Grandidentata, by placing some of the growing leaves inside a transparent box. Like we would have the tree of this plant, we would just pluck the leaves out of it, and then we would place those leaves in the transparent boxes, and then we would try to be calculating the rate of photosynthesis in them. What they have to tell us next is that the researchers measured the uptake of the carbon dioxide by the leaves over a range of temperature from 10 to 40 degrees centigrade. So the temperature they are changing in this investigation. So this would be acting as a variable, and that variable we would say that this is the independent variable. Like the transparent boxes that we have got the leaves of the aspen from the aspen tree, big tooth aspen trees. And on this transparent boxes, we are changing the temperature from 10 to 40 degrees centigrade. And then they say that they carried out their measurements at two different concentration of the carbon dioxide. Like each of the setting would be same, but only the difference would be that at first I would keep the concentration of the carbon dioxide in these transparent boxes having the aspen tree leaves in them at 325 parts per million carbon dioxide, and this is close to what we have got in the external environment. But after providing this much of the carbon dioxide, we would change the temperature from 10 to 40 degrees centigrade. Likewise, we would repeat it up, the same transparent box we would have, and we would label it at J. Now we would keep the concentration of the carbon dioxide at 1935 parts per million, and that they say that this is a very high concentration of the carbon dioxide. Again, the temperature would be fluctuated from 10 to 40 degrees centigrade, and ultimately they would be measuring the rate of the photosynthesis. Now, if we were to simply look on toward the figure 3.1 where they have presented you with the graphs, if you were to see, I have this J in which the carbon dioxide concentration was kept very high. And in H, the carbon dioxide concentration was equivalent to the atmospheric CO2, and this was 325 parts per million. On the x-axis, temperature is coming. This is independent variable that we would change from 0. And now here they have given you the value till 40. But what they have done in the investigation was that the temperature was fluctuated from 10 to 40 degrees centigrade. And here they have shown you the uptake of the carbon dioxide gas. Here, the uptake of the carbon dioxide, this is proportional to the greater rate of photosynthesis. More CO2 uptaken, more would be the rate of photosynthesis. Less CO2 uptaken by the leaves of the great aspen trees that they have presented you above. So ultimately, the rate of the photosynthesis would be less. Now, let us just try to see what the examiner has to ask. For the A part, for the total of 3 marks, they would like you to describe how the results for the aspen leaves that they had in the batch A, they differ from the results for the aspen leaves which they have in the batch H, and they advise you to use the data from figure 3.1 in your answer. So I would just simply revert back to the graph and simply try to look on some descriptive points. Now what you have to be understanding here is that we have been given the value of the temperature on the x-axis, and each of the small box is a representative of 1 degree centigrade, and this is our independent variable. Here we have given on toward the y-axis is uptake of the carbon dioxide in terms of milligram per meter square per minute. So these are the units for the uptake of the carbon dioxide that we are required to be writing in description. And again, you see each small box is representative of 1 unit. So the difference of the J to the H would be that at all the provided temperature that ranges from 10 to 40 degrees centigrade, the uptake of the carbon dioxide of the J is higher. Isn't it like this is higher at all the given temperatures? But if I just look on toward a temperature of 10 degrees centigrade at this point, so the rate of the uptake of the carbon dioxide is same for the leaves that were kept in H and that were kept in J. Moving onward, we would see that the maximum uptake of the carbon dioxide for the J, this was achieved at a higher temperature of 35. But if we just look on toward the H, so the highest carbon dioxide uptake that was taken up by the leaves in H, they got attained at a lower temperature of 25. Then the other point is that the max carbon dioxide concentration uptaken by the J, this is how much? This is 26 milligram per meter square per minute. So this is how the description points would be. Like if I simply just read them up for you, so as I have described it above, that the carbon dioxide uptake in the J is higher than H at all the given temperatures except for 10 degrees centigrade, because at 10 their uptake was the same. And then I have said that the maximum uptake of the carbon dioxide in J is achieved at a higher temperature of 35. Like here, this was attained here at 35. Comparative to H, it attained the maximum carbon dioxide concentration of how much? This was 10 milligram per meter square per minute, but this was attained at a lower temperature. And then the last point would be that the maximum absorption of CO2 in J is higher, and that is 26 milligram per meter square per minute. So this is how total of 3 marks could be attained here. Now moving onward to the next parts, in the B they have to be asking us to be explaining that why the rate of the photosynthesis in the leaves in case of the batch A. So in the batch A, we have the leaves and we need to explain the rate of the photosynthesis. And for 2 marks, you're asking for the first test that why the rate of the photosynthesis increases when the temperature is increased in the range of temperature from 15 to 35 degrees centigrade. So if I just take you back to the graph, for the J, when the temperature is increased from 15 to 35 degrees centigrade, they're asking the explanation that why the uptake of the carbon dioxide or the rate of the photosynthesis is increasing. So what I know is are the two approaches. That when the temperature would be increased, so the kinetic energy of the reactant and the enzymes would increase, and there would be more collisions between the enzyme and the substrate, and ultimately the rate of the photosynthesis would be increased. The other valid point could be that when the temperature is increased, so the uptake of the carbon dioxide would be enhanced. Why? Because an increase in temperature would result in more diffusion of the carbon dioxide into the leaves, and ultimately more carbon dioxide would be used in the photosynthesis, and the rate of the photosynthesis would be increasing. For another 2 marks, what they have to be asking us is that why the rate of the photosynthesis decreases as at the temperature above 35. And if I just look on toward the graph, beyond 35, the rate of photosynthesis in J is decreasing. So the idea here is very simple that why this is decreasing because when the temperature is being increased, so the enzymes would be denatured. And the other valid point could be that with such high temperature, the loss of the water by transpiration would be more. So the plants they would try to reserve the loss of the water, so they would close up the stomata, and by that the CO2 uptake would be reduced. And this is how you would score a total of 2 marks. Moving onward to this C part that they have given in the exam, and this came for the total of 5 marks. And they are saying that you are required to use the results in figure 3.1, and then we need to suggest and explain the likely effect on the plant growth of an increase in the carbon dioxide gas concentration in the atmosphere as a result of the combustion of the fossil fuels. Now, if I just take you back to figure 3.1, so what we see is that when the carbon dioxide concentration was more, in the transparent kind of a chamber that we were given in J. So we have seen that the uptake of the carbon dioxide by the plant was more, and that would mean that the photosynthesis happening in the plant is more. Now, if we just simply try to see what else they have to say is that you have your environment, and we know frequently we have got these fossil fuels, and among the fossil fuels we have the petroleum, we have the natural gas and the coal, and when they would be undergoing combustion, so there would be an increase in the concentration of the carbon dioxide gas in the external environment. And when the CO2 is increasing in the external environment, so that would mean that its uptake into the plants would be greater. And when more of the CO2 would go into the plants or it would be absorbed by the plants, so that would mean that they would photosynthesize more. And by more photosynthesis, they would be making more of the glucose. And once they have made the glucose, so they would respire glucose, and that would release a lot of energy, and by that they would bring about their cell divisions, and that would be increasing their growth. Other than that, we know that for the growth, we need to be making the protein. So when due to an increase in the carbon dioxide by the combustion of fossil fuel, this glucose would be made more, and when this would combine with the nitrate ions, plants would make more of the amino acids and the protein, and that would be again contributing in making new cells and their protoplasm and the cell membranes. The other valid point could be that these glucose they would be polymerized together and they would be used to make the cell wall of the new plant cells. So this is how you are required to be answering it. Now I'm reading my answer for you, and here you would see just 5 statements in order to score a total of 5 marks. So as the answer says that when there is an increase in the carbon dioxide concentration in the atmosphere, so there would be more uptake of the carbon dioxide into the leaves. So this idea is coming from the graph above. And in this way, the plants would photosynthesize more. Why? Because carbon dioxide would no longer be acting as a limiting factor. This would always be available in an excess amount. So the rate of photosynthesis would keep on increasing. And by that, more glucose is formed. And by this respiration, more energy would be released. And that would be used for the cell division. And that would bring about the growth of the plant. By more glucose, we would be able to be polymerizing them, we would be linking them together, and by that we would make cellulose cell wall of the new plant cells. As an alternative point, if you forget any of the one point from here, so you could write in context of the protein making, but you have to firstly combine glucose with the nitrate ions, and then you would make the proteins, and that would make the enzymes or the new protoplasm or the new cell membranes of the plant's cells. Now here come the question again from the leaf structure. And here they say is that we show you figure 7.1, and this is a section through a leaf. We are required to name the structures which are labeled as J and K. Now, if I were to simply highlight again, like you remember is that in the leaves at the top we have the cuticle. This is not being shown here. These are the cells of the peripidermis. These cells that we have here, these are the palisade mesophyll cells. Here we have the spongy mesophyll cells. Again we have the air spaces here, and then there comes the guard cells here. So simply like the spaces they have given you, so we would write for these as palisade cells, and the K that we have been given here, like this is just one guard cell, so we would be writing guard cell here for the total of 2 marks. Now, this is again one of the frequently thing that is being asked from this chapter. For the total of 2 marks, they say that the leaves they carry out the photosynthesis. We are required to write the word equation. Now, here the mistake that the student they do is that if the word equation is asked, they would write the chemical equation. This should not be the way. So for the word equation here, we have written carbon dioxide gas. This is a raw material for photosynthesis, and here comes the water. In the presence of light and chlorophyll, we would end up making the glucose and the oxygen gas out of it. If I were to take you to again May June of 2016, and the variant was 4.1, and this appeared to be the question number 6 of your exam. And for 2 marks they ask us that we are required to state the balanced chemical equation for photosynthesis. So above in the previous question there was that word equation, but now the chemical equation is coming. So that this is a fact 6 molecules of carbon dioxide gas. Now I'm adding plus sign here. We would combine them with 6 molecules of water, and then we are making this glucose. The formula goes as C6H12 and O6, and then along with that we are releasing 6 molecules of the oxygen gas. Now moving onward, here comes the question, and this is an investigation base, and at the same time the examiner would be evaluating our concept of the limiting and the excess. Now what they have to be telling us is that there are students and they are investigating the effect of different wavelengths of the light on the rate of photosynthesis of the water plant, and this is ke boom. Now, the idea is that this wavelength of the light would be changed. This would be my independent variable. And apart from that, the carbon dioxide concentration and the temperature, we need to be keeping them controlled, or we need to keeping them as constant. They have given you the apparatus as well, which the students they use. Now here you see is that we have got this bench lamp. Then we have got this colored filter. By the colored filter, what we mean is that they would be changing it. Like they can be having some red kind of a filter here, green filter here, and by that they would be changing the wavelengths of the light. Okay. Now moving onward, they also have got this funnel, and here they have given you an aquatic plant, and they have added 1% of the sodium hydrogen carbonate solution, and this acts as the source of the carbon dioxide, so we are keeping it constant. The light intensity, we are keeping it constant, but the colored filter that would be changing the wavelengths of the light. Now the oxygen of the gas, it would be released out, and now they are being collected in a gas range. And they have given you a clock here. That means that any of the amount of the oxygen gas that would be collected here in the gas range, they would divide by the total time, and that would give them the rate of the photosynthesis. If we were to look on toward the findings of the student, and they say that the student they collected the gas which was produced by the plant for the total of 5 minutes. So the time is given here, and the gas is being collected. So this is our dependent variable, and the results are shown here. And if we just simply see here, we have got the color of the filter, then there comes the wavelength of the light in terms of the nanometer, and now at the in the end column we have got the volume of the gas collected in terms of cm cube. More volume of this oxygen gas collected would mean that the rate of the photosynthesis was higher, and if the less volume of the oxygen gas would be collected, so that would indicate that the rate of the photosynthesis would be slower. Now, if we simply see like the rate of the photosynthesis in the violet and the blue color filter where violet has got its wavelength of 400, the blue has got the wavelength of 475, the volume of the oxygen gas collected is same. But if I just move onward, you see is that at green filter, where the wavelength of the green color is 550, so the rate of the photosynthesis is the minimum one, and that is 0.20 cm cube only being produced. But again, if we change the color filter from yellow to green and red, and when the wavelength is being increased from 600 to 675, again the rate of the photosynthesis in context of an increase in the volume of the oxygen gas being produced is increasing. So they say is for the total of 3 marks that we are required to describe the effect of the wavelength of light on the rate of the photosynthesis as shown in the student's results in table 6.1, and again you would gain credit if you would quote him the quantitative number from the table. Now first is the general point for description. You would say that as the wavelength of the light increases, the rate of the photosynthesis, initially you see this decreases and then this increases again. Then you would tell him another description point that at the color filters or at the wavelength, like if you see that at the violet and the blue and the red wavelengths of the light, the rate of the photosynthesis is higher. Another description point is that at green filter or at 550 nanometer of the light, the rate of the photosynthesis is minimum. Again, that is 0.20. So these are some of the description points. You can write any three out of it. And I have written the first general point that as the wavelength of the light increases, the rate of the photosynthesis decreases and then increases. This is one general point. And then I would tell him that the rate of the photosynthesis is higher in the violet and in the blue and the red. And then ultimately the rate of the photosynthesis would be slower where? When the filter was green and when the filter was yellow. Alternatively, another description point that I see here is that at the red wavelength of the light or the red color filter where the wavelength is 670 nanometers, so the rate of the photosynthesis is maximum, and then we are required to quote him this value of 0.90 cm cube of oxygen gas being released. So this is how you would score 3 marks for this. Now for 1 mark they say is that state how the student would calculate the rate of the photosynthesis from the results that are shown in the table 6.1. Now you see that the total time for which they carried on the investigation was 5 minutes, and they are collecting or measuring the volume of the oxygen gas being collected in the gas range. So if I'm required to be calculating the rate of photosynthesis, so any of the volume of the gas that we have collected, we will simply divide it by 5 minutes or the unit of time, and then by that we would have our rate of photosynthesis. So simply how you were required to be writing it down, you would say that dividing the volume of the oxygen gas being produced by the total of 5 minutes. Now, if I were to move further for 1 mark, they say state why the student they kept the lamp at the same distance during the investigation. Now, since we were only required to be changing the wavelength of the light, not the light intensity. So by keeping the lamp at the same distance from the plant, aquatic plant that we had in the inverted funnel in the immersed in water, so this would keep the light intensity same. For 1 mark, they again ask us that why we the student they use the sodium hydrogen carbonate solution. So you would say that it provides the carbon dioxide gas. Moving onward to E part, for the total of 3 marks they have said that state three uses in the plant of the carbohydrate which is produced in photosynthesis. Now you see is that by the photosynthesis we would make the glucose, and that glucose it would be used in the respiration. The same glucose would be polymerized together, and by that we would make the cellulose cell wall. The same glucose can be stored in the form of the starch in the chloroplast. The same glucose, when this combines with the fructose, so we make the sucrose sugar out of it. And when the sucrose has been made in the leaves, so this would be loaded into the phloem and it would be translocated to the storage organs or to the sink of the plants. And other than that, the idea is that this glucose would also combine with the nitrate ions, and as a result of that, we would be able to make up the amino acids, and that would further combine to be making the protein out of it. Since the element profile of the glucose and the fats is same, like in the glucose we have carbon, hydrogen, and oxygen, the only thing that we would require would be some specialized enzymes. And when these enzymes they would work on the glucose, so we would make the fats out of them. So any of like three statements you just have to state in three uses of any of these. So I have simply written the first thing is respiration, where the glucose would go into the mitochondria and would be respired. And then I said that it would be used to make the cellulose cell wall. And then other than that, you could say that it can be converted to sucrose or starch or protein. And from the reproduction in plant context, the same carbohydrates they are used to make the nectars. And we know in context of reproduction that it would attract the pollinators with an intention to get their pollen grains pollinated from one plant to another plant. If I were to move beyond, so this again is a question where he would be assessing your knowledge in terms of the leaf structure. And this appeared to come in October, November of 2016. This was variant 4.3, and the exam question was question number 6 with us. They say, name one feature of dicotyledonous leaves that distinguishes them from the monocotyledonous leaves. So you see that in terms of the leaves, what we know is that monocotyledonous leaves, plant leaves, they are narrower, and the dicotyledonous plant leaves, they are broader. Now, this is one difference. Other than that, as an alternative point, you could also say that in the leaves of the dicotyledonous plants, we have got the veins, and these veins they are branching or they are articulated. But in case of the monocots, like if I have got their leaves of the veins, they would be in the parallel manner. So either you say broad leaves are for dicotyledonous, or you could also say that the dicotyledonous leaves they have the reticulated veins. Now moving onward for 1 mark, he asks us that why leaf is an organ. So you would simply say that the leaf is made up of multiple tissues, and we know it is made up of the upper epidermis layer, the lower epidermis layer, the palisade layer, the mesophyll layer. So all of them are the collection of different tissues, and that would make one specific organ, and that is the leaf with us, and this would carry on a specific role of photosynthesis. For 3 marks C, they say that the photosynthesis occurs in the leaf. We are required to be stating the balanced chemical equation for photosynthesis, so it would go as 6 CO2 plus 6 moles of water, and you have to put an arrowhead here, so it would end up making the glucose molecule, and we would release 6 molecules of the oxygen gas. Moving onward, in figure 6.1, which they have given below, so they have shown you a section through a dicotyledonous leaf from a scanning electron microscope. So if I were to simply zoom more, so you see that here we have got the cells of the upper epidermis here. And at this point, we have got the cells of the palisade mesophyll cells, and we see a lot of air spaces here. So this part of the image that is presented here, so this would be indicating the spongy mesophyll cells. But at the same time, this thing that we are being shown here, so this is the vascular bundle with us. And here comes the lower epidermis with us. Now let us just try to see what they have to ask us. And you see very simple thing is coming from the leaf structure. They say that identify the layers which are labeled in 6.1 and explain how their adaptations allow photosynthesis to occur in the leaf. So if I just simply revert back, so you see that the layer J that we have got, this is the palisade mesophyll cells. And what we know is that these palisade mesophyll cells, they are present near to the top surface of the leaf so that they can be capturing more of the sunlight. The other alternative point could be that these palisade mesophyll cells, they have got abundant chloroplasts in them. So that would again allow them to be absorbing maximum of the sunlight. If I were to talk with context to the K, so this K is upper epidermis. And with context to the K, the adaptation would be that they do not have any chloroplast in them. So that means that the light can fall directly to the palisade mesophyll cells. The other idea is that these upper epidermal cells, they are thin or they are flat, so again, more of the light can penetrate through them. If I talk with respect to the L, which is the spongy mesophyll cells, so their adaptation is that they have got abundant of the air spaces in them. So when the carbon dioxide diffuses from the external environment into the air spaces, so alternatively, this can move to the palisade mesophyll cells and the spongy mesophyll cells, and we know CO2 diffusion through these air spaces is very important because without CO2, the glucose cannot be made. As we see in the equation, the CO2 is one of the raw material for photosynthesis. So you simply need to just look on the answer. The layer J for 1 mark, like out of 2, we would say this is palisade mesophyll cell. And for another mark, like what adaptation it has for the photosynthesis, so we would say it has abundant chloroplast to absorb more of the sunlight. The layer K, as we have identified above, this was upper epidermis. And as I've said, either you would say this is transparent or either you would say they are thin. So that would allow the light to pass through them directly to the palisade mesophyll cells. Layer L, you see, we had the spongy mesophyll cells, and their adaptation for the photosynthesis would be that they have the air spaces, and that allows the gases, the CO2, to be diffusing from the air spaces into the spongy and the palisade mesophyll cells, and the oxygen to get out of the palisade and the spongy into the air spaces. Moving onward for the total of 3 marks, but the examiner has to be telling us is that the plant they need nitrate ions for the growth, and they are asking us that why. The reason is that if the plant has to grow, so initially the parent cell they would divide by mitosis to be making new daughter cells, and the new daughter cells they have to make their cell membranes. They have to make, you could say, their enzymes, and all of them they would be made out of the protein. So the usual idea is that when the plant they make the glucose initially in the photosynthesis, so they would combine them with the nitrate ions, and as a result of that we make the proteins, or prior to protein we make the amino acids, and then that protein could be utilized to be making the cell membranes, or it could be used to be making the protoplasm, or it could be used to be making the enzyme, and that is directly linked to the growth. So for 3 marks your answer should be that the nitrate ions they combine with the glucose to make the amino acids, and these amino acids they would further link with one another, and we would make protein out of it. And that proteins they would be utilized for the growth in context of making the new cell membrane, in context of making the cellular organelles, in case of making the transport proteins that we require in the cell membrane, allowing the newly formed cells to function properly. So the last point, it could be written either way. Moving onward, here again is an investigation based question, and this is coming out of the mineral lines, and we know that one of the functional mineral lines that the plant they require is the nitrate ions for making the proteins, the magnesium ion for making the chlorophyll pigment. Now what they have to be telling us is, and this appeared in the May June of 2017, the variant was 4.1, and this was question number 6 of your exam. Now what the student they try to do was to investigate the effect of mineral ion deficiencies on the growth of the radish plant. Okay, so we have the radish plant, and the scientist or the student intentionally they would bring on, or they would be doing some kind of a mineral ion deficiencies, and they would see that how come this is affecting the growth of the radish plants. They say the seeds that were used in the experiment were from the plant that had been self-pollinated for many generations and were therefore all genetically identical. Like the radish plant on which I am doing the investigation, they would be grown from the seeds that would be self-pollinated, and that would mean that all of the plant they would not have any of the genetic variations in them. Now for the 2 marks, what the examiner has to be asking us is that we are required to explain the advantage of using genetically identical radishes in this investigation. Now the idea is that the growth is not only impacted by the mineral iron deficiencies, but this would also be impacted by the genes. Now, since I just want to see the impact of the mineral iron deficiencies on the growth of the radish plant, so I need to be making sure is that only the mineral iron deficiencies need to be seen without any interference from variations in the genomic makeup. So we would say that the advantage of using the genetically identical radishes would be that this would ensure that any difference that would occur in the growth of the plant is only due to the iron deficiencies and not due to the genes, and that would allow us to be making the valid comparison. Now here they are giving us an idea of that how they actually carried on the investigation. Like any of the radish plants that they grew out of the seeds, they divided them into total of 4 groups. Like there was this group 1, group 2, group 3, and group 4. All of them they have the radish seedlings in them, but the composition of the minerals it would be changed in all of the 4 beakers. Like if you were to just simply see here, I have got this mineral iron solution, and then we have got these radishes plants with us. The solution is flowing on the inside, and the solution is also flowing outside. Now for the first investigation that they are doing here, so this mineral ion solution, this would have all the major mineral ions, inclusive of the phosphate and the nitrates and the magnesium ions and all of the stuff. And when they would repeat it for the second time, so they would provide all major ions, but they would not provide the nitrate ions. For the third, they are providing all of the ions, but not the magnesium ions. And for the beaker 4, or for the fourth investigation that they would be repeating. These radishes would be provided with all the major mineral ions, but not the phosphate ions. Now let us move to what the examiner has to ask from all the setting of the experiments which he has presented. Now, if I were to move to this part B, for 3 marks he has asked that state three other environmental factors that could affect the growth of the seedlings. So apart from the mineral ions, we know it very well that the plant they rely on the photosynthesis. So we would say it's the temperature, this is the carbon dioxide concentration, and this is the light that would have an impact on the growth of the seedlings. If I were to just take you to the table here, they have given you this table 6.1, and here they are showing you the result of the investigation which they have performed. Now you see that we have got total of four groups. The group one should be considered as a control group because here we have provided complete mineral lines in their respective solution. The remaining second, third, and fourth, they have got the deficiency of a specific mineral ion. No matter, they have taken different number of the plants in each of the solution, and then they have taken the total dry mass of all the plants in term of milligram in context of their leaves and the roots, and here they have presented the total dry mass, and the total dry mass would mean that if I were to tell you with context to this group number one, so they would pluck their leaves off, they would take the roots of the radish plant, and then they would remove all of the water content out of them, and for them from there they would get the total dry mass. And now here they are calculating the mean dry mass of one plant in terms of milligram. Like for the total of 8 plants, their total dry mass is 2990 milligram, and if I would divide them by 8, so we are getting it for only one plant. If you were to see that the control group having the complete mineral ion solution in it, so their dry mass and their mean dry mass of one plant appears to be greater than the rest of them. I guess the same would go for all the column, like we have to make their comparison with the control group. Now for the C part, they ask for the total of 4 marks that describe and explain the results for the radishes grown without the nitrate ions, and which is group number 2 with us. Now, first I will tell you the descriptive points and then we would require to be explaining them. What I will say is, in terms of description, that the radish plants which are grown without the nitrate ion, their total dry mass of the leaves and the roots is less than the control group. Now, this is one statement, one description that I am making with respect to the total dry mass in context of leaves and roots. The other description point would come that the mean dry mass of the one plant without the nitrate ions, this is less than the group number one, and here we have to take the difference of this value from this value and we would write it down that the radish plant without the nitrates, they have 156 milligram less mean dry mass of one plant comparative to the group number one. Now the explanation is very simple. The radishes which are grown without the nitrate ions, they would not be able to properly make the proteins in them, and without the proteins they are not able to be making the cell membranes, or they would not be able to make the enzymes properly, and that is the reason the growth is less in context of their leaves and the roots, and that is why the total dry mass of group number 2 is decreasing actually. So how you were required to be writing it down, you would say that its growth is less than the control group 1 and having 216 milligram mean dry mass. Like here I am telling him this value, and which has 156 milligram less than the control group. I simply have taken the difference of these just to give my examiner a description point. And then I have like written it in terms of the total dry mass, that the total dry mass of the group 2 for their leaves and for their roots comparative to the group number 1 again is less. Now, this was a description. Now, if I were to talk about the explanation, so I would simply say that without the nitrate ions, the plant in the group 2, they were not able to make enough of the protein, and since the protein is required to make new protoplasm and enzyme, and this has not occurred in group number 2 that was grown without the nitrate ions. Moving onward for 3 marks, for the part D, the examiner asks us that describe the likely appearance of the radish plant grown in the solution without the magnesium ions, which is group number 3, and we are required to be explaining our answer. Now, if we just go back to the table, you see that the plant in the group number 3, they are grown without the magnesium ion, and what you need to be remembering is that the magnesium ions, they are responsible for making the chlorophyll within the chloroplast. So that would mean that since they were grown without the magnesium ion, so the chlorophyll, which is a green photosynthetic pigment, it would not be properly synthesized. So their leaves, they would look yellowish. If you don't want to write it down, then we know it very well that if the chlorophyll is not made within the chloroplast, then they cannot properly photosynthesize, so they would show reduced or they would show stunted growth. So simply like I have just told you, so the answer is that their appearance of the leaf could be yellow. If you don't want to write it down, then you could also write another point that they would have stunted or reduced growth. The explanation would be that the magnesium is required for the synthesis of the chlorophyll and that makes the plant leaves look green. Now for the E part for the 2 marks, they have to telling us is that phosphate ions are the component of the DNA. Now the idea is from the biological molecule, what we know is that the DNA molecule, they have these small repeating units in them. And these small repeating units, they are nucleotides with us. And in the nucleotides, what we have with us are the bases. And these bases, they are technically referred to as the nitrogenous bases that includes adenine, thymine, guanine, and cytosine. Then in one nucleotide, we also have the sugar. And then we have got this phosphate group as well. Okay, so now simply they have told you here that the phosphate ions are the part of the DNA, and actually these phosphate ions they make the small repeating units of the DNA. Now they are asking us that suggest why the radish plants that are available in the group 4 they grew less than the radish plants in the complete solution. Now you know that for the growth the mitosis has to happen where we would have the parent cell at the top and it would divide to be making two identical daughter cells such that the genetic information which the daughter cells would be receiving it should be same as in the parent cell. Now if the plant was not provided with the phosphate ion, so less or no DNA would be produced, and as a result of that, when the cells would divide, so the DNA cannot actually go into the new daughter cells, or the new daughter cells cannot be formed without the DNA duplication. And the DNA duplication can only happen if we would provide them with the phosphate ion and the DNA molecule is sufficient with us. So if I were to just simply read it for you for 2 marks, we would say that without phosphate, less or no DNA is produced, and new DNA is required for the cells to divide by mitosis. That did not happen in this group number 4 radishes plants with us that had the lack of phosphate ions in their solution. If I were to come here to this question number, this other question, and this appeared in October November of 2018, and your variant for the theory was 4.3, and in your exam this was question number 3 with us. And here they say that we show you this figure 3.1, and this is a photo micrograph of a part of the upper surface of the broad bean leaf, which is Vca faba. So this simply is coming from the leaf structure. Okay. Now, if we just simply come on toward the question, they say on figure 3.1, identify and label two structures that are visible in cell P. Now, if you were to simply look on toward the pinot, since this is coming from the leaf, so this is very evident that this kind of a distinct boundary that we see. You can either call this as the cell wall or the cell membrane, the examiner would accept it. And here you see this is a darker kind of a region I see, and this is nucleus with me. So I'm labeling this as a nucleus and I'm labeling this as a cell wall. If you would label it as a cell membrane, it would make no difference. For 1 mark, they ask is state the name of the tissues shown. So you see, these are the epidermis. Why? Because their shape is as such. If they were the palisade, so they would have looked long and cylindrical. If they were spongy, so they would have looked nearly or rough kind of a, you could say, round shape kind of a cells having chloroplast in them. But what I see is their cell wall and the cytoplasm, and they have the nucleus, and we don't see any of the chloroplast in them. So this is for sure the epidermis with us. Moving to this third part, They say that the tissue shown in figure 3.1 is transparent, and we have to explain why it is important to the plant that the tissue shown in figure 3.1 is transparent. Now, if I just take you to the leaf structure, we know at the top we have the cuticle. Immediately below that, we have this epidermis with us, right? And immediately down the cells of the upper epidermis, we have these palisade mesophyll cells. And as far as these palisade mesophyll cells are concerned, so what we know from our prior knowledge is that they have these abundant chloroplasts in them. So you see, when these upper epidermis cells, when they would be transparent, so this means that the light would not be blocked and it would fall directly to the palisade mesophyll cells, allowing their chloroplasts to absorb more of the sunlight to photosynthesize more, making more of the glucose. So for 3 marks, we would simply say that since these are transparent, so it would not block the light, but rather than it would allow the light to pass through them. And then they would be absorbed by the chloroplast of the palisade cells, and this would allow them to carry more of the photosynthetic process to be making more of the glucose. So you see here there are the three statements in terms of explanation for us to be scoring total of 3 marks here. Now for another 3 mark in part B, they say is that the stomata they are found on the lower surface of the broad bean leaves, and they want us to describe the function of stomata. Now you know that on the lower side of the leaf, what we have with us are the cells of the lower epidermis, and in between them we have these guard cells, and in between the guard cells we have the stomata. Now from here we are getting an idea is that during the daytime, these stomata they would allow the carbon dioxide gas to diffuse into the air spaces of the spongy mesophyll cells, and at the same time they would also allow the oxygen gas to escape out of the leaves into the external environment. So by this, these stomata they are controlling the exchange of the gases. Okay, the other idea is that here in the spongy mesophyll cells, where we don't only have a lot of air spaces, but at the same time we have got these xylem vessels, and we also have these phloem vessels. So during the daytime, from the xylem vessels or from the cell wall of the spongy mesophyll cell, the water would evaporate here, and with the help of the transpiration, these water molecules they would diffuse into the external environment, and that would create a pull for the water to be pulled upward into the leaves through the xylem vessels. Now this again has to be regulated, because if we are not controlling the amount of the water that is going into the external environment by the transpiration, so the plants they would have a deficiency of the water and they would wilt. This is again something I don't want. So you see that how much of the water should escape into the external environment by the transpiration. This again is under the impact of the stomatas. When the external temperature or the wind speed is high, so these stomata they close up, and by that they would minimize the loss of the water by the transpiration. So for 3 marks, how your answer should look like that these stomata they control the exchange of the gases that has to be exchanged between the leaves and their absolute external environment. During the day, they would allow the carbon dioxide to diffuse in for photosynthesis and would allow the oxygen to diffuse out of the leaves. And as of now, third point would be that they control the loss of the water by the transpiration process. If I were to take you to this table, they say more than 40 years ago, botanists are the specialized people, those who deal with the study of the plants, and they studied the leaves of the broad bean plants and discovered that the guard cells control the opening and the closing of stomata. They found that stomata were open when the guard cells were turgid. Simply, they are giving us an idea that when these botanists, when they studied the working of the guard cells, what they believe is that when these guard cells, when they would fill up with the water and when they would become turgid, so by these guard cells becoming turgid, the stomata that we have in between them, they would open up. And now table 3.1, which they have presented us, they give us some of their measurements. Now here we have the closed stomata and then we have the open stomata, right? And what I see is that when the iron concentration in the guard cells it is zero point 0.3, so the guard cell volume is 4000, and the turgor pressure in the guard cells is 2.0, and the width of the stomatal opening is zero point 0. So this means that when the guard cell volume is less, so this means water has not moved into the guard cells, and that is why the stomatas are closed. But on the other way around, when the stomatas they have to be opening up, so what is happening? That the ion concentration in these guard cells is increasing, and that would bring more water into the guard cells, and that would for sure be increasing the turgid pressure in the guard cells. And that is why the width of the stomatal opening in terms of the micrometer is increasing. So this means that the elaboration which they gave us at the top, this is right. Now, for the 2 marks, what they have to be asking is that the ions they move into the guard cells by active transport. Now they want us to describe that how the ions move into the guard cells. So the idea is very simple that when we look on toward the guard cells, so we have their cell wall. Immediately below their cell wall, we have their membrane, and that membrane is the cell membrane. And within the cell membrane, we have got these transport proteins, and these transport proteins, they are the carrier proteins, right? So, what would happen actually is that within the guard cells, we have the mitochondria, and these mitochondria they would release energy by the respiration process, and that would help in the movement of the charged particle or the ions from their lower concentration on the outside of the guard cells to their higher concentration on the inside of the guard cells, and that would be happening with the help of the carrier protein. So this is the entire mechanism by how the ions would be moved into the guard cells. So for 2 mark, you would simply say that these ions, by the active transport, they are moved against the concentration gradient because they would be moved from the lower to the higher concentration, and for that, the energy would be released from the respiration process. And these ions, they would come on the inside of the guard cells with the help of these carrier proteins. Now, for another 5 mark, they want us to be describing and explaining that how the change in the ion concentration causes the guard cell volume to change, and we are required to use the information in table 3.1 in our answer. So what I have described the data above, that when the stomatas, when they are open, so the ion concentration inside of the guard cell is a higher value, and that is 2.5 p parts per mole or p per mole, and that would have resulted to be decreasing the water potential inside the guard cells, and that water would go on the inside by the osmosis, and that is why the guard cells when they would become turgid. So the width of the stomatal opening in terms of description, we would say this is 8.0 micrometer. If we were to be looking on toward the explanation is that once the ions they have come on the inside of the guard cells, so automatically the water potential in the guard cells would become lower. So the water would move into the guard cells by osmosis. Their large vacuoles would increase in size. They would exert the pressure on the cell wall. The cell wall would bear this pressure exerted by the large vacuole, and by that they would become turgid. So for 5 mark, how your answer should have looked like, we would say that when the stomatas are open, the ion concentration in the guard cell is 2.5 p per mole, and which is higher when they are flaccid, like in the first column, in the second column, which they have shown us. We would say that when the ion concentration is higher in the guard cell, so this would lower the water potential inside of them. Okay, in the stomata comparative to the external environment. Like here, we should be changing the word. This word should be the not the stomatas, but rather than it should go as guard cells. Like when the ion concentration has increased on the inside of the guard cell, so for sure this would lower the water potential on the outside of the guard cells comparative to their external environment. And in this way, the water would move into them by osmosis across a partially permeable membrane. And when the volume of the guard cells increases, so this creates a push on their cell wall, and by that the guard cells would become turgid and the stomatas would be compelled to open up. So this is how you would score total of 5 marks for this. If I were to move further for 2 marks, they say is that these botanists they left the broad bean plants unattended for 3 days. Like for 3 days, they did not bother about these broad bean plants, and they said that during this time the broad bean plants have wilted. Now they want us to suggest two environmental factors that causes the plant to wilt. So why the plants they wilt? That could be either due to the speed of the wind. Okay, speed of the wind is actually a scenario like I have these guard cells, and on the outside, if I just simply increase the speed of the wind, so any of the water vapor that are escaping with the help of the transpiration, so they would be carried away fast, and that would result in more loss of the water by transpiration. And the only way around is for the plant to wilt so that the loss of the water should be minimized. The other scenario is that if by some mean or the other, if in the external environment, if the humidity is decreasing, so the water content or the water vapor content in the external environment would be less. So that would again result in the more loss of water by the transpiration, and that would again cause the plants to wilt. Since it was with context to the external environment, so I have said it in terms of the wind and the low humidity in the air. You could also say the temperature as well, because in the external environment, when the temperature is higher, so any of the water that is being escaping by the transpiration or by the diffusion, they would have a greater kinetic energy, so they would be moved fast, and again the plant would wilt. So moving onward, here we have got another question from May June of 2019. This was your theory paper variant 4.1, and this was question number 2 of your exam. And this would be from the factors that limit the rate of the photosynthesis, where you have learned about that the limiting factors of the photosynthesis is carbon dioxide concentration, the temperature, and the light intensity. So, what they have to be telling us initially is that the rate of the photosynthesis of terrestrial plant can be determined by measuring the uptake of the carbon dioxide. This is like the right approach. More CO2 that would be taken up by the plant, that would indicate higher rate of photosynthesis, because we know carbon dioxide gets used up in the photosynthesis, and when this combines with the water, so more of the glucose would be made. Now, for the total of 2 marks, they want us to explain why the plants take up carbon dioxide during photosynthesis. So we know that carbon dioxide is a raw material for the process of photosynthesis. And when the concentration of the carbon dioxide is higher in the external environment, so it would cause the CO2 to diffuse into the plants, where the CO2 later would combine with the water to be making the glucose and the oxygen gas. Now, if we were to just come here, and they are telling us about an apparatus, and they would tell us that how come this would be utilized, and they have to be telling us is that the rate of photosynthesis of the parts of individual leaves can be measured using a handheld device as shown in figure 2.1. Like if you were to see here, like this is the part of the plant where this handheld device has been kept. So this apparatus would allow us to measure the uptake of the CO2 at this point, and by that we can see that how much of the photosynthesis is happening at this part of the leaf that they have shown us in the diagram. Now, in context of this handheld apparatus, what they tell us is that this apparatus allows the air to flow through the transparent chamber that encloses part of the leaf. Like this is the enclosed part of the leaf, and this transparent chamber would allow the air to flow through it. And the apparatus would then measure the carbon dioxide concentration of their entering and leaving. Like at this point, how much of the CO2 is entering and how much of the carbon dioxide is escaping. So that idea would be given us by this transparent chamber, and for sure there would be some kind of readings that would come here that the person would read. Now for 2 marks, they want us to explain that how the results from the apparatus can be used to calculate the rate of photosynthesis. Above they have said that the uptake of this carbon dioxide gas, this gives us an idea in context of the rate of photosynthesis. So what I need to be doing here is that I would calculate the difference between the amount of the carbon dioxide gas that entered through the transparent chamber and that left through the transparent chamber, and then I have to be dividing it by time to calculate at how much of the carbon dioxide this would be taken up per unit of the time, and that would be proportional to an increase or a decrease in the rate of photosynthesis. If I were to take you to this part, they say that there is a student and he used the same apparatus which was shown above in figure 2.1 to investigate the effect of the temperature on the rate of the photosynthesis of the leaves of the Chinese plantain. The genus is Plantago and the species is Asiatica at two different of the carbon dioxide concentration A and B. Now overall, our independent variable that we have to be changing here is the temperature. But the temperature that we would change, would have to be making sure is that we would vary the concentration of the carbon dioxide That would be A and B. And the figure 2.1 below, this shows us the result of the investigation. Like if I were to take you here for the graph, so you see that the temperature of the leaves is being changed from 10 to 40. And here they have given you the rate of photosynthesis in term of micromole per meter square per second. That is the rate of photosynthesis being shown here. Now, for the A, you see that the carbon dioxide concentration is 370 parts per million. And for the B, the carbon dioxide concentration is 1000 parts per million. But I have to assume from here is that no matter if the concentration of the carbon dioxide is low or high, with an increase in the temperature of the leaves, the rate of the photosynthesis is increasing. However, for the curve A which is shown here, so you see that since the carbon dioxide is provided in a less amount or in a controlled amount, so at this point our temperature would become excess, but the carbon dioxide concentration and the temperature they would become limiting here. And that is why when we are increasing the carbon dioxide concentration from 370 to 1000 parts per million, so you see the rate of photosynthesis is increasing and this is not leveling off like this. Why? Because the CO2 is not becoming limiting. So the rate of photosynthesis is being shown as it continues to increase. Now here the ask is that we are required to state one environmental factor that should have been kept constant in this investigation. Like what we see in the graph is that they have fluctuated the temperature readings from 10 to 40, and then they are also changing the concentration of the carbon dioxide as well. So the third external factor that has to be controlled during the investigation, this would be the light intensity. Now, for the 3 marks, they want us to describe the effect of the temperature on the rate of photosynthesis when the carbon dioxide concentration A was applied. So now the first point would be the basic point. What you have to be saying in terms of description for A is that as the temperature increases, the rate of the photosynthesis increases. But at 30 degrees centigrade, the rate of the photosynthesis being attained, this is reaching a maximum value of 22 micromole per meter square per second. But when the temperature is increased beyond 30 and up till going till here till you see 35 degrees centigrade temperature. So here the rate of photosynthesis levels off, okay, beyond 30 degrees centigrade temperature. So you know it well from the previous questions as well, that how we are required to be writing in terms of description. So the first is the general point that as the temperature increases, the rate of the photosynthesis increases and then levels off. And the rate of photosynthesis increases from 12 micromole per meter square per second to 22 micromole per meter square per second from 10 to 30. Like simply from 10 to 30, I have quote him this value. This is being increased from here and then coming till here. And then beyond 30 degrees centigrade, the level of the rate of the photosynthesis, it would level off or it would attain a plateau or this would become constant at 22 micromole per meter square per second. Now moving on to this third part, here the examiner wants you to be calculating the percentage increase and that too in the rate of the photosynthesis, and he has given you the value of the temperature which is 30 degrees centigrade, and he asks this that you must calculate it when the carbon dioxide concentration was increased from A to B as shown in figure 2.2, and at the same time this is clearly written that our final answer should be to the nearest whole number. Now, if I move back to the graph, now you see is that here the temperature that we have been given, this is 30. And from this, I would come on toward the curve A and B. So when we come on to curve A at for 30 degrees centigrade here, the rate of the photosynthesis that we are given here. So this is 22 micromole per meter square per second. Likewise, I have to go on toward the curve B because you see that when the CO2 concentration was increased from this value to this value, so rate of the photosynthesis, it significantly increased. So I have to come to this curve and then I need to go on toward the y-axis. So here the final value that we have for the rate of the photosynthesis is 30 micromole per meter square per second. So this is simply what we require the values and now we need to substitute it into the formula. So as I have written here already, at 30 degrees centigrade for the curve A, it is 22. For the curve B, this is 30. And now for the percentage increase, here is the final value minus initial value. Then we need to divide it by the initial value, and we are required to be multiplying it with 100 because this is a percentage increase that we are required to be calculating. So 30 minus 22 divided by 22, and when we multiplied it by 100, so the answer that we are getting is 36.36. But since it is to the nearest whole number, so when we just simply like round it off to the nearest whole number, so we would write it as 36%. So this simply indicates that when you are increasing the carbon dioxide concentration from A to B, so the percentage increase in the rate of the photosynthesis is 36%. Now, if we come on toward the fourth part, they say explain the effect of increasing the temperature on the rate of photosynthesis for the carbon dioxide concentration. And for 3 marks, examiner wants you to also be using the term limiting factor in your answer. So let me just take you back to the graph again, and you see is that at any of the point we don't see this curve leveling off, or this is reaching a plateau. So this would indicate us that at all the values of the temperature that we are given, temperature is acting as a limiting factor. The carbon dioxide gas cannot act as a limiting factor because we have provided a very huge concentration of the CO2. And why the rate of the photosynthesis is increasing with an increase in temperature? The reason is that the increase in temperature would not only impact the working of the enzymes, but at the same time, this would also be increasing the diffusion of the carbon dioxide gas into the leaves so that more of the CO2 could be moving in and ultimately resulting in a greater rate of the photosynthesis. So if I were to just simply read the answer for you, as I have explained above, that for the curve given in the B, the temperature is a limiting factor over the whole provided range because the plateau is not leveling off. And we would say that when the temperature is increased, the diffusion of the carbon dioxide into the leaves would increase, and at the same time, the temperature would also impact the enzymatic reaction because when the temperature is increased, so the average kinetic energy of the enzyme and the substrate would increase. So their frequency of the collision would increase, and for sure, we would make more enzyme substrate complexes out of it. And that is why the rate of photosynthesis in the curve B was seen to be increasing and not leveling off. Now, moving onward, for 1 mark, they are asking that the student concluded that the carbon dioxide concentration is the factor limiting the rate of photosynthesis between 30 and 35 degrees centigrade for the results shown for A in figure 2.2. And then he says is that you are required to provide the evidence for this conclusion. So now, if I just take you back to the curve A, the kind of the evidence that we see is that beyond 30 degrees centigrade and coming till 35 degrees centigrade, despite we have got a lot of temperature with us, so the temperature is not acting as a limiting factor here, is basically there in excess. So for sure, this is a low amount of the carbon dioxide now from this point onward where this is leveling off, that this is actually the CO2 which is limiting the rate of photosynthesis or is causing the curve to be leveling off. Now, if I were to read my answer for you, so you see, I have written that the rate of the photosynthesis tend to increase as the CO2 concentration increases, but until at 30 to 35 where it levels off, and that would indicate that temperature at 30 between 35 is more, but the CO2 concentration is something that is leveling it off. Now, likewise, if we were to move to part D, they say that the similar investigation was again carried out on Arizona honey sweet. So this is again another plant. The genus is Tidystromia and the species is Oblongifolia, and they are grown where? In the Death Valley in California. And over there, the highest temperature may be greater than 45 degrees centigrade. So you see that if we just see this investigation that was being performed, so for here the temperature range is given from 10 to 40 degrees centigrade. But the type of the plant which they are talking about now, in this part, what we notice is that here the temperature can go beyond 45 degrees centigrade. So what I see is that here the rate of the photosynthesis max is coming till 45, and beyond this 45 degree centigrade temperature, the curve does not seem to rise. Like I don't see any further rise in the rate of the photosynthesis, which is given on toward the y-axis. Now, what he asks for the total of 2 marks is that we are required to predict and explain what would happen to the rate of photosynthesis if this investigation is continued at a temperature higher than 45. So if I would I would increase the temperature for this investigation, and that temperature is greater than 45. So what I know is that since the photosynthetic process is under the influence of the enzymes, and with so much higher temperature, the enzyme would lose the shape of its active site. The active site would deform, and as a result of that, it would not collide with its substrate molecule, resulting in less enzyme substrate complex formation, and that is why we would see a decrease in the rate of photosynthetic process. So what we can say here is that the rate of the photosynthesis, our prediction is that it would decrease at such a higher temperature. The reason is that the enzyme's active site they would begin to denature, and their binding with the substrate would decrease. As an alternative valid point, what we can think else of is that when the temperature is so much high, above 45 degrees centigrade, so the plants they would start conserving the water, so that they would close their stomatas. And when the stomatas would be closed, so the CO2 uptake into the leaves would decline, and that would again result in a lower rate of photosynthesis. Moving onward to the next question, and that is again from the external factors that limits the rate of photosynthesis, and this appeared to come in the May-June of 2019. The variant for your theory paper was 4.2, and it was question number 2 with us. What the examiner has to be telling us is that in the recent years, the concentration of the atmospheric carbon dioxide gas has increased. And for 3 marks, they want us to be telling them or to be describing them that what were the possible causes of increased atmospheric carbon dioxide. Now, the idea is very simple that he wants us to be telling him the ways, the possible causes that have resulted in an increased carbon dioxide concentration. So you know that whenever we would have these fossil fuels and when we bring about their excessive combustion in the automobiles, like for the gasoline or if the coal is being burned, so this would lead to an increased carbon dioxide in the environment. The other possible cause is the deforestation because we know that the plants they use the carbon dioxide in the photosynthetic process, and if we are bringing about the deforestation, so the number of trees would reduce, and with less trees meaning is that less CO2 would be absorbed from the external environment. The last point that we can quote him is that this can also be due to an increase in the human population because we human beings continuously are respiring, and with an increase in the number of the people would mean that there are now more people respiring CO2 gas into the external environment. Moving onward, here comes the B part in terms of the investigation. And what they did, that they took the soybean plant, the genus was glycine, the species was max, and they were grown in total of two separate plots. And for each of the plots in which they were growing these plants, so they used an enrichment system. And by the enrichment system, what we mean is that they would be using some burners. And by that, they would be able to be controlling the atmospheric carbon dioxide concentration for the plants that they were trying to grow. What they have to be telling us next is that for the plants that we have in the two plots, the atmospheric carbon dioxide that was varied. For the first plot, the carbon dioxide concentration kept was 370 parts per million, and that is equivalent to the current atmospheric carbon dioxide concentration that we have now. For the second plot, the carbon dioxide that they are using is a bit higher, and this is 550 parts per million, And this concentration of the carbon dioxide is equal to the possible future atmospheric carbon dioxide concentration, like what can happen in the future in terms of the CO2 that could be available. Now, after these soybean plants, they were fully grown. So what the scientists did, that they calculated the average rate of the photosynthesis at regular interval from 4 a.m. to 22 p.m. for both plots. Now, since we are changing the CO2 concentration, that would be our independent variable, and we are measuring or calculating the average rate of photosynthesis, so this would be our dependent variable. Now, if we were to be looking on toward the results of this investigation, on the x-axis they have given us the time of the day, and on the y-axis we have been given the average rate of the photosynthesis in terms of micromole per meter square per second. Let us move forward to see what the examiner has to ask. So this was total of 6 marks, and here the examiner wants you to describe and along with that to explain the effects of the carbon dioxide concentration and how this was affecting the average rate of photosynthesis of the soybean plant from 4 a.m. to 22 p.m. And here they have clearly given you the instruction that you are required to use the data from figure 2.1 in your answer. Now let us just revert back to the graph for the description and the explanation points. Again, 6 marks, 3 points should go for description, and then we have to be giving their explanation. What you are required to be noticing is that although they carried their investigation from 4 a.m. till 22 p.m., but I don't see any of the values before 6 a.m. and beyond 20 p.m. Right? So we just are given the values from 6 a.m. and then ending till 20 p.m. So let us just track that what description points do we have with us. What I see is that at 12 p.m. the rate of the photosynthesis is maximum for 370 part per million carbon dioxide concentration and for 550 parts per million carbon dioxide concentration. Because the explanation is that at 12 p.m. the light intensity at midday, this is actually the midday where the light intensity is the max, so it would not be acting as a limiting factor for the rate of photosynthesis. This would be excess. And that is why this is accounting for the peak rate of photosynthesis for this and this. And in description of this point, we are required to be quote him that at 12 p.m. the maximum rate of photosynthesis is 22 micromole per meter square. And for 370, this is nearly 15.5. So this was the description and explanation for this. The other point that I see here is that the photosynthetic process, it starts at 6 a.m. and then it ends at 20 p.m. And this is the common sense because at 6 a.m. the sun would try to emit the light radiation, then at 20 p.m. that would be 8 p.m. actually. So here there would be no light available with us. So that would mean the explanation would be that at the start of the day at 6 a.m. and at the end of the day at 20 p.m., since the light would be acting as a limiting factor for photosynthesis, here literally no light would be available, so the photosynthetic process nearly stops, like becomes zero. And at this, the graph keeps on going, so this means that the light has started to emerge out of the sun, and that is why the graph they appear to go above. So this was the explanation for it. The other description that I have to be presenting him is that for this concentration of the carbon dioxide, the rate of photosynthesis at all the given time is more comparative to the lower graph. And the explanation is very simple because when we would have a greater carbon dioxide concentration, so its diffusion into the leaves would be more comparative to this. So that would result for a higher rate of photosynthesis for this curve comparative to this curve. Like I would simply would like you to now just have a look on my answer. As I have said that the rate of photosynthesis for both concentration of CO2, they peak at 12 p.m. and reaching 22 micromole per meter square per second for 550 parts per million CO2 concentration and 15.5 micromole per meter square per second at 370 parts per million carbon dioxide concentration. This is description. And the explanation is that at this part of the day, which is 12 p.m., the light available for photosynthesis is maximum and it would not limit the photosynthetic process. The other description point that I indicated above was that the photosynthesis it starts at 6 hours and stops at 22 hours. And why? Because light is required for photosynthetic process and at these times it would be acting as a limiting factor because the light would be less available at 6 and it would nearly be zero at 22 hours. So our photosynthetic process would become zero. And the rate of photosynthesis, as I said, at 550 part per million of the carbon dioxide concentration, this was higher at all the given time than this much concentration of CO2. The reason is that at higher concentration of the CO2, which was 550 parts per million, there would be more diffusion of the CO2 into the leaves, resulting for more photosynthesis than 330 part per million carbon dioxide concentration. Now, moving onward to its C part, that where the examiner would be evaluating your approach in terms of the leaf structure. And they say the scientists they also made observations of the leaf structure of the soybean plant. And by the leaf structure, we know that it has cuticle, the upper epidermis, the lower epidermis, the palisade mesophyll cells, the spongy mesophills, and the stuff like that. They say epidermis and the mesophyll tissues are adapted for photosynthesis. Now, here he has used the general words. He is neither saying upper or lower, he is neither saying palisade or the spongy mesophyll, but rather than generalized, they are asking. They want us to complete the table 2.1, where we are required to state two structural features of each of these tissues and explain how each of the feature is an adaptation for the photosynthesis. So what I know is that in the epidermis, if this is the upper epidermis, so ultimately it would be transparent and it would lack the chloroplast. Like either you could write transparent or you could write it lacks the chloroplast. And how this would be an adaptation for the photosynthesis would be that it would allow the light to pass directly through the mesophyll cells to the mesophyll cells. Now, in the epidermis, what we know is that in the lower epidermis, there are guard cells, or you could say there are stomatas, and they allow for the diffusion of the carbon dioxide gas into the leaves. Moving onward to the mesophyll, I can be having one point that I would take from the palisade mesophyll cells, other point that I would take from the air spaces that we have in the spongy mesophyll cells. So in context of the palisade mesophyll cells that are a part of a mesophyll tissues, I have written that many chloroplasts and they trap more of the sunlight. The other point would be the presence of the air spaces and they allow more movement of the gases between the cells and those air spaces that we have with us. Now, by this, we would score a total of 4 marks. Now, if we were to come on its last part, which is D, and for the total of 2, he is asking that when the scientists they were working in the plot with the carbon dioxide concentration of 550 parts per million, you remember at the top there were the plants that were grown in this much higher concentration of the CO2, and when the scientists they were working, so their breathing rates they were higher comparative to when they were working in that plot where the carbon dioxide concentration was less. And they're asking that why their breathing rates got higher in this concentration of the carbon dioxide gas plots. Now we know as that in these plots where the CO2 is higher, so it would be detected by the brain. And that brain, it would send more of the electrical impulses to the intercostal muscles. And by that, it would be increasing their breathing rate. Because by working in this environment, they would be inhaling more of the CO2 into their body, and the carbon dioxide concentration is increasing in their blood plasma, so they would like to get rid of it. And that is why the body works in a way that it would bring about an increase in breathing rate for them. Now, for this question that came in the May June of 2019, and this was variant 4, 3, and for the exam, this was question number 2 with us. For the first part, the examiner is interested to know that whether you know the word equation for photosynthesis or no. Now, this is a word equation for 2 marks, and what we know is that we require the carbon dioxide gas, it has to combine with the water, And as a result of that, we are required to be making glucose as one of the product of the photosynthesis and the oxygen gas as the other product of the photosynthesis. For the B part, again, this there would be a kind of a situation where you would be evaluated at whether you are clear about your concept of external factor of photosynthesis or not. Now, this says that the scientists they investigated the effect of the light intensity on the rate of photosynthesis in the leaves of eucalyptus trees, and that too at two different concentrations of the carbon dioxide A and B. Now, the idea here is that we would be changing the light intensity, and that is our original independent variable. And further, when we would be doing this investigation on the leaves of this tree, so throughout the investigation, we would have a different concentration of carbon dioxide A and that too in case of the B. And they say that the results are shown in figure 2.1. So if we were to just come on toward the graph, on the x-axis they have given you the light intensity in terms of the arbitrary unit. And toward the y-axis they have given you the rate of photosynthesis. Now anything that comes on toward the x-axis, this is our independent variable, and whatsoever comes on toward the y-axis, this is our dependent variable. So we are measuring this and we are changing the light intensity. Now, what they have to be giving us is a key here, and they say is that the curve A that you see in the graph, here the carbon dioxide concentration was 140 parts per million, and for the B curve, the carbon dioxide concentration was 1000 parts per million. So there and then you should come above, and you should be labeling this curve to avoid any confusion. Before I try to give you an idea that how you're required to describe or explain this graph that is presented in this figure 2.1. Let us just go to the question to see what the examiner has to ask. So for the first part, for 2 marks, he is asking us that you are required to suggest and explain why the scientists kept the temperature of the leaves at 20 degrees centigrade while they recorded the result. Now, what your approach should be that the external factor that can be affecting the rate of photosynthesis, they are the temperature, it's the light intensity, and the carbon dioxide gas concentration. Now, since in this investigation we are overall looking on toward the effect of the light intensity, so the light intensity should change. And if we would allow the temperature to be changing, so alone we cannot evaluate the effect of light intensity alone. The other idea is that the temperature has a direct impact on the diffusion of gases, and this can also affect the enzymes that contribute in the photosynthetic process. So that is why the temperature has to be kept constant. Now I have told you some 3 or 4 points. Now you have to write any two out of it. The first point that you could be telling your examiner is that temperature is a factor and it directly affects the rate of photosynthesis. Hence, we need to standardize it. So we can alone see the effect of changing the light intensity. If you are not interested to be writing these points, you can tell your examiner that any increase or the decrease in temperature can affect the enzymes or it can also have an impact on the diffusion of gases. So this is something we don't want. Now here comes the second part from the mathematical calculation. For the total of 3 marks, what he is asking is that you are required to calculate the percentage increase in the rate of photosynthesis at a light intensity of 1250 arbitrary unit when the carbon dioxide concentration was increased from lower 140 parts per million to 1000 parts per million. What I am required to be doing is that I would go back to the curve and I would see from the x-axis this value of 1250, and then I would go to the curve for 140 part per million and 1000 part per million, and we will try to be seeing is that what was the percentage increase in the rate of photosynthesis owing to an increase in carbon dioxide concentration from this to this. And the examiner is simply saying that your answer should be to the nearest whole number. Okay, so let us just move back. So here you see that here we have got the value of the light intensity, which is 1250. And from here, I would go to the curve of A, and then I would come on toward the y-axis because here we are given the rate of photosynthesis, and you see here the value that we have with us. So this is 21, 22, and 23 micromole per meter square per second. Now, when the carbon dioxide is being increased to 1000 part per million, so I would go to this curve now and I would see that how much photosynthetic process has been increased. So this is 40 micromole per meter square per second. I need to take the difference of these two values and then I'm required to be dividing it by the initial value. Then I would multiply it with the 100%. Automatically, I would get the answer in term of percentage increase. So you see, I have already written it down that the rate of photosynthesis at 1250 arbitrary unit of the light intensity for 140 part per million, this was 23 micromole per meter square per second. And at 1000 part per million, this was 40 micromole meter square per second. Now, here I am putting the final value and I am subtracting the initial value. Then I am dividing it by the initial value. Then we are required to be multiplying it with 100. The answer coming is 73.91%. So when I would round it off to the nearest whole number, this would become 74%. Now, for this part, what he asks is that describe the effect of increasing light intensity on the rate of photosynthesis when the concentration of the carbon dioxide was 140 parts per million. So we will revert back to the graph and you will see that for the part of the graph where the carbon dioxide concentration kept was 140 parts per million, how come this has impacted the rate of photosynthesis? And again, I would say for description, you are required to be writing whatsoever you see in the graph. So if I just revert back to the graph which they've given us, so you see is that the curve A that we are given here the carbon dioxide concentration kept was 140. So what I see is that as the light intensity is increasing, so the rate of the photosynthesis that I see on toward the y-axis, so this also increases and then it levels off. Now, this is the first description point. For the second description point, you just try to be finding out the point where you see the maximum photosynthetic rate. So I see it for the x-axis. This was at 1750 arbitrary unit of the light intensity and the maximum rate of the photosynthesis that I see here. So this is 25 and we are required to be writing the unit and we would say this is 25 micromole per meter square per second. The other point for the description is that an increase in the rate of the photosynthesis is more steeper where the light intensity values are less. So this is what you were required to be writing in the answer. So if I just take you to the answer that I have written as per the mark scheme, so I said that as the light intensity increases, the rate of photosynthesis increases and then it levels off. The other point was that the maximum rate of photosynthesis it was reached at 750 arbitrary unit of the light intensity and the value for this was 25 micromole meter square per second. And then I said that the increase in the rate of photosynthesis this is steeper at the low light intensity. Moving onward to the next part, this is part 4 of this question. And for the total of 4, what the examiner has to be asking is that you are required to be explaining the effect of increasing light intensity on the rate of photosynthesis when the concentration of the carbon dioxide was 1000 parts per million, and he is clearly asking you to be using the term limiting factor in your answer. So against 4 mark we are required to be giving 4 reasons in order to be scoring total of full marks. So we would revert back to the graph and we will see that how come the light intensity has impacted the rate of photosynthesis when the carbon dioxide concentration kept was 1000 parts per million. So you are required to be seeing the graph again. Now this is a curve, that is curve B, and in this part the carbon dioxide concentration kept was 1000 parts per million. So what I can assume is that earlier in A, when the carbon dioxide concentration was kept so minimum at 140 parts per million, so at this point, the light intensity was becoming excess and the CO2 was becoming limiting and it leveled off. But what I see is that for the B, as the light intensity is increasing, so does the rate of photosynthesis. And the graph is not leveling off at any of the given values of the light intensity. So what you have to be noticing at this point is that I am given the light intensity on my x-axis and this is the independent variable. And the independent variable would always remain limiting before the plateau is reached. So for B, what I see is that nowhere the plateau is being reached. So that would mean that the light intensity is remaining limiting throughout the values ranging from here to here against this much higher concentration of the carbon dioxide gas. And you see that we have increased the carbon dioxide to such a maximum value that this CO2 concentration would remain excess. That is why the graph at any point is not leveling off. What you would be writing here is that at 1000 part per million carbon dioxide concentration, the rate of the photosynthesis would increase with increasing light intensity. And the other statement would be that the light intensity remains limiting at all the provided range of the light intensity. And the reason is that the graph is not leveling off at any point. For the third mark, what we would say is that since the carbon dioxide gas provided is so much high, so it remains excess throughout and would result in more of the diffusion of the CO2 into the leaves and it would not become limiting, rather than it would remain excess. The other valid point could be, the fourth point would be that when you have increased the light intensity so much, and we know that this light energy would provide the chemical energy for the photosynthetic process, and that is the reason with increasing light intensity at 1000 part per million of the carbon dioxide gas, the rate of the photosynthesis, it intends to increase. So I would now simply read my answer for you as per the mark scheme. So we would say that at the carbon dioxide concentration of 1000 part per million, the rate of photosynthesis is increasing with an increasing light intensity. And the other point would be light intensity remains limiting at all given values because the rate of photosynthesis does not level off even at higher values of the light intensity. And then what we would say next is that with highest carbon dioxide concentration that you people have provided, and that too at 1000 part per million, so throughout the investigation, the CO2 would remain excess and would not become limiting. For the fourth mark, we would say is that with higher light intensity, more light energy is absorbed by the chloroplast of the plant, and that light energy would convert into the chemical energy, and that is something that would be carrying out the photosynthetic process. Moving on toward the next question, and this again is from the external factor that limits the rate of photosynthesis. It came in the May-June of 2021. The variant for your theory paper was 4.3, and this was question number 3 in your exam. An idea of the investigation which the examiner has presented in figure 3.1. This has to be clearly understood so that we can answer accurately. What they have to be telling us is that there is a scientist and he is measuring the rate of photosynthesis, and that too in the leaves of the sunflower plant. The genus of that plant was Helianthus anus. Further, they say is that the scientists, they use apparatus which is shown in figure 3.1 to measure the rate of photosynthesis. Now, since in the statement we are given the word rate, and if I have to be assessing the rate of the photosynthesis, so what I know is that in photosynthesis, 6 molecules of the carbon dioxide, they combine with the 6 molecule of the water, and as a result of that, we would make 1 mole of the glucose, and we would also release 6 moles or 6 molecules of the oxygen gas. Now, whenever the word rate is coming, so this means that we have to relate it to the time. And if I wanted to calculate the rate of photosynthesis, what I can do is that in how many time the amount of the CO2 was utilized, or the other way around could be that how much volume of the oxygen gas was released with respect to the time. So this that would give me the rate of photosynthesis. Now, if you were to clearly see here, the examiner has placed the plant, and this is coming in the chamber 2. And again, we have got this chamber 1. Now, in chamber 1, this is attached to an airflow regulator. And to this chamber 1, they have also attached some kind of a temperature probe so that they can keep an eye on that the temperature is being kept constant or not. And what we can see is that the carbon dioxide gas at a concentration of 400 parts per million, this is going into the chamber 1 where the temperature kept was 20 degrees centigrade. For sure, the CO2 would now come into the chamber 2 and it would be diffusing into the leaves where it would be used up in the process of photosynthesis. Now, any of the unused carbon dioxide gas, it would come into the chamber 3, and the scientists here, they are calculating as that how many carbon dioxide has remained unused. And what I see is that we provided this plant with initial concentration of carbon dioxide at 400 parts per million, and the CO2 that is getting out of the chamber 3, this has got reduced to 360 parts per million. So my idea would be that the total amount of the CO2 getting used up is 400 minus 360. So the plant has used total of 40 parts per million carbon dioxide concentration. And if I would divide this value with time, so I would get the rate of the photosynthesis in term of CO2 or in term of the reactants being used up per unit of the time. Now for the A part, what the examiner has to be asking is that the apparatus in figure 3.1, it maintains a constant temperature and a constant humidity. Why they are maintaining the temperature and the humidity? Because they can have an impact on the photosynthetic process. The temperature would directly impact the enzymatic reactions that are responsible for the photosynthetic process, and the humidity has something to do with closing and opening the stomata. So now if we just see for the 4 marks, they asked us that why the temperature has an effect on the rate of photosynthesis. So what we know is that this rate of the photosynthesis, this is directly controlled by the enzymes. And as we would increase the temperature, so as a result of that, the kinetic energy of the enzyme would increase and the substrate would also increase. And that would bring about more of the collisions between the enzyme and the reactant molecules, and that would lead toward more enzyme substrate complex formation. And as a result of that, we would see an increase in the photosynthetic process. Now here you would score 4 marks. I have written 4 points for the explanation. Now if you wanted to be writing some alternative points, so what we know is that when you would be changing the temperature or you would be increasing the temperature, so that would result in more diffusion of the carbon dioxide into the leaves. Again, another alternative point could be that if you would increase the temperature beyond limits, or if you would provide very high temperature, so this could also be denaturing the enzymes. Okay, so this is an exact explanation, and if you would like to skip some points out of this, and you can also add these points as well. So here, this is how you would score total of 4 marks. If I were to move further, for 3 marks, what he has to be asking is that explain why the rate of photosynthesis will decrease if the humidity in the apparatus becomes low. So here we have to be relating a decrease in the humidity to how come this is affecting the rate of the photosynthesis. Now, if you were to recall the structure of the leaf, then we know is that we have got this lower epidermis layer. And in the lower epidermis layer, what we have with us, these are the guard cells. And in between the guard cells, we have this opening coming here. And the name of this opening, what we know is that this is stomata. Usually this so happens as That during the daytime, in order to pull the water up the xylem from the stem and the roots, so the water would escape out by the transpiration process. Now, the transpiration depends on the temperature, on the speed of wind, and on the humidity. Now, if I were to be lowering the humidity in the external environment, so this would mean that the amount of the water molecules that we have got in the external environment, this would also be lowering down. So now more of the water would begin to be lost in the transpiration process. But the plants, they are smart, so they would have to avoid this because the plant is relying on the water for its support, its rigidity. So as a strategy, what the plant would do at this point is that they would be closing the stomatas. The guard cells would become flaccid and the stomatas would be closing up in order to be avoiding more loss of the water by the transpiration. But that would be an advantage in terms of reserving the water. But the closure of the stomata would have a disadvantage as well, that here the carbon dioxide gas diffusion into the leaf, it would stop, and the carbon dioxide would become limiting now, and ultimately the rate of the photosynthesis, it would decrease now. So for 3 marks, what you are required to be writing was that when the humidity is low, the loss of the water by transpiration process, it increases. And to avoid this and to conserve the water, the guard cells, they become flaccid and these stomatas, they become closed up. And that would reduce the diffusion of the carbon dioxide into the leaves, and automatically, without the CO2, the photosynthetic process cannot be continued at the higher rates. If I were to move to this third point, and they would ask for 2 marks, is that the apparatus was left for the total of 15 minutes. And they want you to explain how the scientists would use the readings for the concentration of the carbon dioxide in the chamber 1 and the chamber 3 to calculate the rate of photosynthesis. And I have explained this at the start of this question, that we would take the difference in the amount of the carbon dioxide gas that we are supplying to the chamber 1 and what remains in the chamber 3. So initially 400 parts per million of the CO2 is going and 360 remains unused, and this is getting out. So this means that this plant right here in the chamber 2, this used total of 40 part per million carbon dioxide. And now this is the CO2 that got used up, that is 40 parts per million. If I were to calculate the rate out of the reactant, which is CO2, so I have to be dividing it by the total time, and that presented here was 15 minutes. So how you would write is that the difference in the carbon dioxide concentration, it needs to be calculated between the carbon dioxide that is entering into the chamber 1 and that is leaving through the chamber 3. And then we need to divide this carbon dioxide being used up by the total time of the 15 minutes, and this is how we would calculate the rate of photosynthesis. Now, let us just come here and try to see what the examiner has to present here for its P part. They tell you is that the scientist, he carried out another investigation using the same apparatus but at different temperatures. Like earlier, if you remember, the temperature was kept constant, but now he is doing the same thing. He would be calculating the rate of the photosynthesis by looking on toward at how much of the carbon dioxide is getting used up with respect to the time, but here he would start to be changing the temperature. So that would mean is that for this investigation, this temperature change, it would be acting as our independent variable. Now they say is that they measured the rate of uptake of the carbon dioxide in the light, and then they measured the rate of release of carbon dioxide in the dark. Now we know it very well that anything that we measure up, that is our dependent variable. And in this investigation, when the temperature would be changed, so our dependent variable would be that how much of the carbon dioxide would be taken up in the light when they would be photosynthesizing. And they also measured that how much of the release of the carbon dioxide would be happening in the dark. Now here the idea is that why come the carbon dioxide is uptaken, or this is being used up in the light. So the answer is very simple, that in the light the plants are photosynthesizing, so they would uptake the carbon dioxide. But when we talk about the dark, so we know that in the dark the photosynthesis would stop and the plant they would be respiring. Now they have given you this figure 3.2 and they are showing you the results of this. Now, if you just look on toward the x-axis, this is temperature in terms of centigrade, so this is our independent variable. And on toward the y-axis, they have given you the rate of carbon dioxide uptake and release. Uptake in the light and release in the dark in terms of microgram per hour per centimeter square of the leaf. Now you were to see here in the graph, if I just indicate it, that with an increase in temperature, the carbon dioxide uptake in the light, this is remaining a bit constant here, or there is a very slight decline. And ultimately from 20 degrees centigrade onward, the CO2 uptake in the light, this is decreasing. And on the other way around, when the temperature is being increased, so if you just look on toward the curve indicated with the dotted line, so the carbon dioxide being released in the dark, this is increasing. Okay, so this is simply I am elaborating the graph that is given here. At this point, you should not waste your time in thinking that why the CO2 uptake in the light is decreasing with temperature and the CO2 release in the dark is increasing with temperature. You first go on toward the question to see what the examiner has to ask. Now, here for 2 marks, what he has asked is that use information that is presented in figure 3.2, and by that you are required to complete table 3.1. Now, the temperature is 20 degrees centigrade and they want us to calculate the rate of uptake of the carbon dioxide. The units are already given here, so you are never required to be writing units here, else you would lose some marks. So let us just revert back to the curve to the curve, and we look for the temperature of 20 degrees centigrade, and I have to go on to the curve where the CO2 was being uptaken. So you see, as here is a 20 degrees centigrade temperature. From here I am coming toward the curve where we see the carbon dioxide uptake in the light, and you see from here I am coming to this part of the graph. Now you notice as that in between 140 and 120 we are given 5 small boxes, so each box would be representative of 4 units. So here from 20 I am coming here, and the uptake is 124, 128, 132, and this is 136. Okay, so that is why we are writing 136 here. And the next he says is that you have to give us a value at the same temperature of 20 for the rate of release of carbon dioxide. Now, again, I have this temperature of 20. I come on toward the curve where the CO2 is being released, this dotted one, and now I go on toward the y-axis, and here the value that we see as this is 20. So we have simply written up 20 here. Now here for 2 marks, this was a little technical but not difficult. The scientist determined that photosynthesis in the leaves at 35 degrees centigrade used carbon dioxide at a rate of 135 microgram per hour per centimeter square of the leaf. And from figure 3.2, what this says that this shows that the rate of carbon dioxide uptake at 35 degrees centigrade was 90 microgram per hour per centimeter square of the leaf. Now this is something little tricky. Now you see as that in the lower epidermis we have the guard cells, and in between the guard cells we have the stomatas. At this temperature of the 35 degrees centigrade, what we notice is that plant is taking up the total amount of the carbon dioxide, that is 90 microgram per hour per centimeter square of the leaf. But originally within the leaves, the carbon dioxide being used up for the photosynthesis is higher. Like see, there is a point. Like CO2 diffusing in is 90, but within the plants, the CO2 being used up within the chloroplast, this is higher. This is 135. So that would mean that against the uptake, that is 90.
So the plants are using more CO2, so that would imply that where does that increased CO2 is coming from, if this is not coming from the external environment. And we see a very major difference, like the difference is 135 plus 10, and that is 45 micrograms. So here we have to be asking us that from where is this excessive or the extra amount of 45 micrograms coming, even though the plant was absorbing only 90 microgram. So the reason is that in addition to this CO2 which is diffusing into the plants for the photosynthetic process, what we know is that the plants they also have the mitochondria. These mitochondria they would be releasing the carbon dioxide gas, and instead of diffusing out during the day, this CO2 would start getting used up in the chloroplast. So this extra 45, you could say, microgram of the carbon dioxide, this is being supplemented from the respiration that is happening within the mitochondria. So that is why they ask us for 2 mark that explain why the rate at which carbon dioxide is used in photosynthesis is greater than the rate of the carbon dioxide uptake. So in the photosynthesis, more CO2 is getting used up and less CO2 is taken from the external environment. And answer would be that the plants they respire at all the time and they would release CO2 as a result. And that would start getting used up in the respiration together with this amount of the CO2 which they are taking up from the external environment. Moving onward for 1 mark, what they have to be asking is that explain why the results in table 3.1 they are expressed as per centimeter square of the leaf rather than per leaf. So the CO2 getting used up, they calculated that per centimeter square of the leaf, how much CO2 was used not in terms of an entire leaf. The idea would be that in this investigation, the size of the leaves could vary. So when we would calculate the carbon dioxide being used up per centimeter square of the leaf, so this would provide us with the valid comparison. So the answer here that you were required to be writing was that the leaves they have different surface area. So when we would express the carbon dioxide being used up per centimeter square, so this would allow for making the valid comparison. Moving onward, so this question again is from the external factors limiting the rate of photosynthesis. It appeared in the May-June of 2022. The theory variant was 4.2. This was question number 1. And for your exam, the part C, D and E, it was from this chapter of photosynthesis or the plant nutrition. What they have to be telling us is that figure 1.1, it shows a spongy mesophyll cell from the leaf of the plant. What we know is that leaf is an organ, it has the cuticle, the upper epidermis, the palisade, and the spongy mesophyll cells. We also have the lower epidermis, in them we have the guard cells, and we also have the stomatas. And from that leaf, they have given you the diagram of the spongy mesophyll cell. They say the arrow shows the net direction of the movement of the carbon dioxide molecule during the daylight. Like this is the sun. Outside there is daylight. So in what direction the net movement of the CO2 is happening. For this figure 1.1, what I see on the outside is the cell wall. This is cell membrane. Here we have this large vacuole. Here we have the nucleus. This is mitochondria. This is chloroplast. So the CO2 is going from the mitochondria to the chloroplast. As we know, these mitochondria, they respire, they would produce CO2, and this is carried to the chloroplast. At the same time, you see is that outside of the spongy mesophyll cells, we would have the air spaces. And out of these air spaces, the carbon dioxide is also going to these chloroplasts. If I were to move onward to see what the examiner was to ask from this, so against 1 mark, he is asking us that carbon dioxide is a raw material for photosynthesis. They want you to state the process by which carbon dioxide travels into the leaves from the air, so we know this is a diffusion process. For 2 mark, the examiner wants you to describe the pathway which is taken by the molecule of carbon dioxide from the air outside a leaf to a spongy mesophyll cell. So we are required to be writing the pathway for the molecule of CO2 that lies in the air outside. So you see is that the CO2 would diffuse, it would pass through the stomata that lies in between the two guard cells, and eventually this would come into the air spaces that we have in the spongy mesophyll layer, and from there this is diffusing into the cells of spongy mesophyll. So simply you would write it as that it enters through the stomatas that is present between the guard cells, and then it goes into the air spaces which are available within the spongy mesophyll cells, and then ultimately it moves into the individual cells that we have got here. Moving on toward the D part, they say is that the lower concentration of the carbon dioxide in the air may restrict the rate of photosynthesis in the plant. So I am lowering the CO2 in the air, and this directly is impacting the rate of photosynthesis. What they have to be asking was that state the term given to something which is present in the environment in such a short supply that it restricts the life processes. So the life process that is under the impact of the CO2 is the photosynthetic process. And if it gets in short supply, so the technical term that we use for this is a limiting reactant. Moving onward, they say is that state one other feature of the environment that may also restrict the rate of photosynthesis. What we know is that there are three fundamental external factors that can limit the rate of photosynthesis. That is the carbon dioxide concentration, that is the light intensity, and that is the temperature. So now other than the CO2 they were asking, so I would say that low, or you could say no light intensity, and that can also act as a limiting factor. For E part, the total marks were 2, and the sad is that the researchers have devised a process of artificial photosynthesis, and for that they used gold nanoparticles. They act as a catalyst, and they utilize the green light to convert carbon dioxide to the fuels such as propane. Now, this is such an astonishing kind of the findings that they brought into play. Like what we know is that the propane originally is extracted from the fossil fuels, and there is a lot of load on the fossil fuels when we would obtain propane out of it. So, what the scientists did that they take the use of the gold nanoparticles, and they cause these gold nanoparticles to convert this green light, and by that the carbon dioxide in the external environment would be converted to a fuel. So now they want you to suggest advantages to the environment of using artificial photosynthesis on a large scale. So now since we have got this fuel that we are making from the CO2 in the external environment, so that would lead to the conservation of the reserves of the fossil fuels. As of now, there is a great load. We are using more of petroleum or the coal or the natural gas to be obtaining this propane. But once we do it with this way, so the load on the fossil fuels will reduce. Now, since this process is also using the carbon dioxide gas, and we know carbon dioxide is a greenhouse gas, and an increase in the amount of the carbon dioxide is resulting in global warming. So when this would be starting using up for this process, so the amount of the CO2, which is a greenhouse gas, so it would decrease in the environment, and the problem of the global warming can be minimized to some extent. Moving onward, we have this next question, and this is again from the external factors of the photosynthesis. It appeared in the May-June of 2022. The variant for your theory paper was 4.3, and this was question number 5 with us. Against 3 marks, what the examiner has to be asked, telling us is that the glass houses they are designed to maximize crop planting, and this is a kind of an isolated environment where the plants they are grown to maximize their yield. What they intend to be asking is that explain why the carbon dioxide enrichment is used in many glass houses to increase the crop plant yield. And by the word carbon dioxide enrichment, what we have to be thinking is that in these glass houses, we would keep the carbon dioxide concentration more by the artificial processes. What we need to be thinking at this point is that this carbon dioxide is one of the factors that can limit the rate of the photosynthesis. So when I would be providing more of the carbon dioxide by the enrichment process by burning the coal or the natural gas burners, so the amount of CO2 in the glass houses would remain more, it would diffuse more into the leaves, and it would not become as a limiting factor for the rate of photosynthesis. So how the answer should look like that the carbon dioxide gas is a limiting factor of photosynthesis. The amount of the carbon dioxide gas which is present in the environment is limited, so it can become less and can limit the rate of photosynthesis. When we bring about the carbon dioxide gas enrichment in the glass houses, so it would remain excess and it would not limit the rate of the photosynthetic process, and more CO2 when it would go into the leaves, more photosynthetic process would happen, and the plants they would make more of the glucose that would allow for their maximum growth. For the second part, he has to be asking us that suggests how the carbon dioxide concentration in the glass houses can be enriched, it can be increased. So the idea is very simple. Either we would burn the coal with the help of the burner, or we can use natural gas burners, and by that the CO2 in the glass house would become enriched. Against 2 marks, they ask you is that outline how the carbon dioxide in the glass house moves into the leaves. So we know outside of the leaves, the carbon dioxide gas is higher in concentration. So it would diffuse from that higher concentration in the glass house, and by that it would pass the stomata that lie in between the guard cells. Now coming toward this B part, they further has to be telling us is that there are the countries and they have the temperate climates. And in those temperate climate countries, what they are doing is that they are providing additional lighting again so that it should not become limiting. They have given you table 5.1 where they have summarized some of the factors that are considered by the plant growers when choosing the type of the lamps to install in the glass houses. Like there are many types of the lamps that are available in the market. Now, which type of the lamp they would install in their glass houses? So it is depending on various factors, like how much of the electricity the lamp is consuming, and against that consumption of the electricity, how much of the light intensity is being emitted out, and they are giving you the notes of these lamps as well, that what are the advantages of each and every type of the lamp that we have got. Now we should not be wasting our time in thinking that what is best suited among them. We would have to go on toward the question first. For 2 marks, this is mathematical calculation, and they intend to be asking us that calculate the percentage increase in the energy used by the metal halide lamp compared to the energy used by the fluorescent lamp, and our answer it should be near to two significant figures. Now, if I just revert back to the table, they are indicating about the metal halide lamp, and at the same time, they are indicating us about the fluorescent lamp. Metal halide lamp and its electricity consumption, this is 651, and when comparative to the fluorescent lamp, so this is 394. And we have to be calculating the percentage increase for the energy used by this metal halide comparative to this fluorescent lamp. So you see as that I have clearly written that the energy used by the metal halide is 651 joules per second, and by the fluorescent lamp this is 394 joules per second. For the percentage increase, the final value is bigger, the initial value is smaller. We are required to be dividing it by the initial value of 394. Since percentage has to be calculated, so I am multiplying it with 100. The answer coming is 65.228%. And they said to the two significant figures. So these 65 alone is up to two significant figures. So we would write it as 65%. Now against 1 mark, they ask is that which type of the lamp has highest light intensity output per unit of the energy they are using. Now here the idea is that if I talk about the sodium, they are asking is that against the electrical energy they are using in terms of joules per second, how much is the light intensity output out of it? And we have to be considering that that is producing more highest light intensity against electrical energy used up. So for the sodium, if I were to tell, against 1041, the light intensity emitted out, this is 1767. So here we have to be employing as the unitary method. So simply I would say that against 1041 joules per second, the light intensity being used up is 1767. So against 1 joules per second, how much would be used? So simply we would divide this 1767 value by 1041 and we would get an answer out of it. So I have done the same. This value, I divided it by their electrical energy use up value and I have got the values. And here we would see that way we are getting the maximum value. And the maximum value is coming for the sodium. That against 1 joule per second of the electrical energy being used up, the light intensity being emitted out is the highest, which is 1.69. So that is why we are writing sodium here. Moving onward, for 3 marks, they say some types of limbs, they release a lot of heat and they are asking the possible effect of the excessive heat on the plants in the glass house. Wherever we have to be relating the heat, excessive heat in this case, and its impact on the plant, so we just need to think in terms of enzymes. And what we know is that the photosynthetic process, this involves the enzyme. And if we would use some of the lamps that emits out a lot of heat or excessive heat, so for sure it would denature the enzymes in the leaves or in the cells of the leaves. And by the result of that, the shape of enzyme's active site is lost. And at the same time, what we can think other than this enzymatic approach is that the transpiration or the loss of water by transpiration is also dependent on the temperature. So when there are the lamps that are emitting a lot of excessive heat, so the plant's transpiration would increase and this can also cause the plant to wilt. So this is how you would score a total of 3 marks for this. Moving onward to this question, and this happened to appear in the February-March of 2023. The variant for your theory paper, this was 4-2. And in that paper, this was question number 4. The B and the C part, they were related to the photosynthesis, and in this, we would see something relative to be writing the equation of the photosynthesis and the use of the glucose that would be there in some other part of the plant that would be carried from the leaves. They say figure 4.1, they show you a photograph of a tree that has been girdled. And the girdled mean that they are removing some part of the stem out of here. And usually when we do the girdling, so the phloem vessels they are removed. They say that girdling involves removing a complete circle of the bark and phloem from around the tree. So if we would remove the phloem from this part of the plant, so after some time this part of the stem it would start to appear swollen up because any of the sucrose that is made in the leaves from the glucose, it would come till this point in the phloem, but it cannot go further beyond this point. They have to be asking us for 3 marks says that explain why the glucose made during photosynthesis is required for the absorption of the mineral ions by the roots. Now, if I were to record the root hair cell, so you see they have the cell wall and they have the cell membranes. And what we know is that in their cell membrane, there would be availability of some transport proteins and the carrier proteins. The root hair cells, they have to be absorbing some of the charged particles from the soil, and in the soil, the concentration of the charged particle is less. If I talk about the large vacuole that is available within the root hair cells, so here the concentration of the charged particles comparative to the outer soil, this is higher. So here lies a lower concentration of the ions or mineral ions. Within the large vacuole, the concentration of these ions is already higher. So if the root hair cells are required to be absorbing these ions against the concentration gradient with the help of the active transport, so what we know is that the energy has to be released from their mitochondria. And the mitochondria, they can release energy by the respiration only when they are provided with the glucose. So that is why the glucose has to be provided for the active transport to take place, and that would move the ions against the concentration gradient, and that is why there is necessity of the glucose. So how the answer should appear that the absorption of the ions by the roots occur by the active transport. And by that, the ions that we are talking about, they are moved against the concentration gradient. And this protocol would require the use up of the glucose in the respiration process within the mitochondria of the root hair cells that would provide the energy for this active transport process to happen. Now here comes very typical equation for the 2 mass where we are required to be writing balanced symbol equation for photosynthesis. And you see is that here we have got this carbon dioxide gas, this is the water, this is glucose, this is oxygen, and we have to be putting 6 with CO2, water, and oxygen gas. So this becomes balanced symbol equation. They did not say word equation, symbol mean in terms of the chemical symbols. Now, if we just come here, this again is from the external factor of the photosynthesis. This happened to appear in the May-June of 2023. The theory paper variant was 4.1, and this was question number 4 for your exam. They have to be telling us is that this is this figure 4.1, and this shows the effect of light intensity on the rate of photosynthesis. So the light intensity effect we are changing, this is our independent variable. And not only that they are changing the light intensity, but they are carrying it at different temperatures and the concentration of the carbon dioxide gas. Now, overall, the light intensity is coming on toward the x-axis. This is my independent variable. And they carried out 3 investigations as A, B, and C. And here on the y-axis, they are giving you the rate of the photosynthesis. So this would be our dependent variable. When they carried the investigation for the A, temperature was 25, CO2 concentration was this much. For B, temperature was lower. This was 15 degrees centigrade. The carbon dioxide concentration was 0.4%. For C, the temperature is again kept at 15 and the carbon dioxide concentration is 0.04%. Here is a trick. If I just consider about C, before the graph can reach a plateau, like at this part of the graph, these would be excess and the light intensity would be limiting. But at this point where this is becoming horizontal or this is reaching the plateau, so the temperature and the carbon dioxide, they would become limiting and the light intensity would become excess. Now, if we talk about the curve B which the examiner has presented here, so the point where the graph is leveling off, like from this point onward. So you see is that here the carbon dioxide gas that we have and the light intensity that we have, they would become excess, but the temperature that is just kept as in the previous investigation that is C. So this becomes the limiting. If I just talk about this part of the curve where this is not leveled off. So here you see the light intensity is limiting, but the temperature and CO2 they would be excess. But beyond this point onward, the CO2 becomes excess, the light intensity again become excess, but the temperature it would become limiting from this point onward. If we talk about the A curve, temperature is further increased. Now since this was becoming limiting in B, so now when I'm increasing it, the rate of photosynthesis intend to increase more. So that would imply that the point where the graph is leveling off, here none of the light intensity or the temperature or the carbon dioxide is limiting, all of them they're acting as an excess. So this means that there could be some other reason for this, for the photosynthetic process being leveling off. Because when I do not have the limitation of any of the external factor, so at this point, the enzymes that are responsible for carrying out the photosynthetic process, they have reached their maximum. So we can't further increase the photosynthetic process beyond this point. I have given you the elaboration of this curve. Now let us just try to see what the examiner has to ask. This was challenging. For 6 marks, we are required to describe and explain the reasons for the shape of the line B and the C that we have got. Now, I would be reading these points and I would be indicating what we were required to be writing here. We would say in terms of description that as the light intensity increases, the rate of photosynthesis for both B and the C, they rises initially, see, like they are rising initially. But in each rising part of the curve, no matter we have B or C, so the light intensity in the rising part of the curve C and in the rising curve of the part B, so this is acting as a limiting factor. And we have to be explaining this that light provides energy which is needed for the photosynthetic process. Now moving onward, we see another thing that at higher light intensities, the graph for B and C, this levels off. Why? Because for C, the light intensity becomes excess here. For B, the light intensity again becomes excess here. But for C, the carbon dioxide becomes limiting. And for B, the temperature becomes limiting. So that is why we would say as that each line for B and C, this levels off at high light intensity. So that would indicate that light is no longer the limiting factor. There would be some other factor which would limit the photosynthesis. If I talk about the line B, where the temperature is this much and the carbon dioxide is this much that I have written in the brackets here, so we reach the maximum photosynthetic rate than C. Like for B, it is higher comparative to C. And here the reason was that the B comparative to C, this has higher carbon dioxide concentration. So more CO2 would be able to diffuse in the leaves for photosynthesis. And the line C that we see, this levels off earlier, like this is leveling off here. But for the B, the leveling off the rate of the photosynthesis is at a higher light intensity. So this is a description point. So as is written here, that for the line C, where the temperature is this much and CO2 concentration is this much, so this levels off at a lower rate and does so at lower light intensity. Okay, like this is leveling off at a lower rate and also at a lower light intensity because the reason is that at lower light intensity it would be acting as a limiting and the concentration of the CO2 for the curve C is since less comparative to this, so this would become as a limiting factor, and that is why the rate of photosynthesis cannot be increased beyond this point onward. So this is how you would score total of 6 marks for this. Now for 1 mark, they ask you is that glucose is one of the product of photosynthesis. They are saying that state the chemical formula of the other product. Now the chemical formula mean I would never write oxygen gas. I will write the formula for this. For 4 marks, they want you to outline how the carbohydrate made during photosynthesis are used in plants. Now the first thing is that the glucose would be used up in respiration to release energy. Then I can also polymerize the glucose or I can link them together and I can convert it to the starch for the storage process. Glucose at the same time combines with the fructose to make sucrose. And the sucrose we know can be carried for the translocation process with the help of the phloem vessels. Now, here you would score total of 4 marks. But if you were to be forgetting any 1 point from above, the alternative bullet point would be that these glucose at the same time can polymerize to make the cellulose, which is a component of the cell wall, the glucose and the fructose that are made in photosynthesis. So this again can be used to make the nectars. And further what we know is that the carbohydrate inclusive of the glucose, when I would combine it with the nitrate, I can make also the proteins out of it. The composition of the carbohydrate and the fats in terms of the element which they possess is same. Both have carbon, hydrogen, and oxygen. So once we make the glucose in the photosynthetic process, we just need to be employing some of the enzymes, and they can also convert the carbohydrates into the fats. So I have written 7. You can write any 4 out of it. Moving onward to this question. This came in the October-November of 2023. The theory paper variant was 4.2 and this appeared to be question number 4. And again, It's from the limiting factor of photosynthesis. We have got the student and he investigated the impact or the effect of temperature, that too on the rate of photosynthesis in one species of the plant. The temperature would be changing, so it would be independent variable. The rate of the photosynthesis we would be measuring, so this can act as a dependent variable. Further, they have to be telling you is that the photosynthesis involve enzymes controlled reaction. So this is a hint here that some elaboration is going to come in the question in terms of your enzymes elaboration. Further, they say is that we have got the leaves, and from the leaves we are cutting the discs out of them, and we are keeping them at different temperatures. In order to be for the surface area of the discs to be kept constant, because this would act as a standard variable. So this surface area of the leaf was kept constant for each of the temperature change that they were bringing about by. Now, the rate of the photosynthesis which they are measuring is in terms of the volume of the oxygen gas that would be produced by each of the leaf disc. So this would be the dependent variable. That is measuring the rate of photosynthesis, but in terms of the volume of the oxygen gas being released out. And that would give us an estimate of the rate of photosynthesis. Now they have presented you the results in 4.1. Temperature comes on toward the x-axis. This is independent. Here we have rate of photosynthesis. In terms of arbitrary unit on the y-axis, this is our dependent variable. Now they want you to be answering them as that identify the optimum temperature for photosynthesis in figure 4.1. So the optimum temperature would be where the rate of the photosynthesis is maximum. And I see it at 40 degrees centigrade. And here the rate of photosynthesis reaches 37 arbitrary unit, because in between this and this we have got total of 5 units. So each small box is representative of 1 unit. So here this would be 36 and here we have this 37. That is in terms of maximum rate of photosynthesis, that is 37, but the optimum temperature that we have here, this is 40 degrees centigrade. Moving beyond, for 6 marks, they say is explain the results shown in figure 4.1. They are not asking the description, they are asking the explanation for the curve. What I see is that when the temperature is being increased from 25 to 40, the rate of the photosynthesis increases, right? And the reason behind this would be that since this photosynthetic process is under the impact of the enzymatic activity, so when we would be increasing the temperature, the kinetic energy of the substrate and the enzymes, it would increase, they would collide more enzyme substrate complex would be formed about by. However, when we are increasing the temperature beyond 40, so you see the rate of photosynthesis does not become zero, but this is declining, this is decreasing, because beyond 40, all those attractive forces that maintains a three-dimensional shape of the enzyme molecule, they start to break off and the enzyme molecule would start to be losing the shape of the active site, and by that the collision of the substrate within the enzyme's active site, it would decrease, leading to less enzyme substrate complex formation, and that would be identical to a decrease in the rate of photosynthesis. So for 6 marks, how your answer should look like, you need to give 6 points for this. That when the temperature is increased from this to this, the kinetic energy of the enzyme and the substrate molecule would increase. And that would bring about more frequency of the collision between the enzyme and the substrate. And we would make more enzyme substrate complexes now. At 40, at the optimum temperature we see is that there would be maximum number of enzyme substrate complex that would be formed. But when the temperature would be increasing beyond 40, so the enzyme's active site would start to denature and it would no longer remain complementary to the shape of the substrate, and that is why beyond 40, the curve for the rate of photosynthesis, this was going downward. So I hinted you earlier that since they said here themselves that photosynthesis involves enzyme controlled reaction, so this was a hint that would help you in writing the answer. Now, for 2 marks, they say carbon dioxide was supplied in excess at each temperature during the investigation. Since we are looking on toward the effect of the temperature on the rate of photosynthesis, and I also know that the carbon dioxide and the light intensity, they are also some of the external factors that can impact the rate of photosynthesis, right? So, in order to see that this is only the temperature which is impacting the rate of photosynthesis in this investigation, the carbon dioxide has to be supplying in excess so that it should not become limiting. Because if this would become limiting, then ultimately that impact of the temperature on the rate of photosynthesis cannot be studied alone or separately. So for 2 mark, how this would be written as that this would always keep the CO2 as an excess and this would not become limiting. And this would make us sure that any effect or impact that we are getting on the rate of photosynthesis is only caused by a temperature change, not by CO2. For 1 mark, they say is suggest why not all the oxygen produced by the leaves is released. Now, the plants they are making the glucose and the oxygen gas in their chloroplast for their own mitochondria. This is only during the day that the O2 released from the chloroplast is in excess. So it would not be entirely used up by the mitochondria. So that is why excess is getting released out into the external environment. So why not all the oxygen produced by the leaf is being released? Because the cells of the plant itself, they would use some of the oxygen gas in their respiration process. Now for 2 marks, they have to be asking us that describe the role of photosynthesis. So we know as that the chlorophylls are the green photosynthetic pigment and they would trap the light energy and then would convert it into the chemical energy. And that would be utilized to be making the glucose or you could say the carbohydrates. And by this, you would score 2 marks. Let's crack the next question. And this is from your elaboration of the leaf structure. Appeared in October-November of 2023. The theory variant was 4.3, and this was question number 2, A part for your exam. And they say is that this figure 2.1 is a diagram of the cross section of the part of the leaf. Now, what I know is at the top we have the cuticle, here we have the cells of the upper epidermis, here we have got the palisade mesophyll cells, then here I have got the spongy mesophyll cells. This is spongy mesophyll cell. Here we have the air spaces and this guy right here that we see. So this is a vascular bundle. The big boxes that you see here, they are the xylem and the bottom one that we see, they are the phloem. Here, these guys right here, these are the guard cells and the opening among them, this is stomata. For 4 marks, they say is that identify and explain how the structure labeled J and K they are adapted for photosynthesis. So if we just talk about J, so they are the cells of the upper epidermis. What we know is that they lack the chloroplast, and that is why they would allow the light to pass directly to the palisade mesophyll cells who have the abundant chloroplast in them so they can photosynthesize at a maximum rate. So this is how the answer would look like that this is upper epidermis cell. They are transparent as there is no chloroplast present in them and this allows for the maximum light to pass directly to these palisade cells. As I have highlighted earlier that the K are the xylem vessels and we know that they are continuous or you could say they are hollow or you could say they lack cytoplasm or you could also say that they have no cross bonds like any one word from here and that allows them to carry water and ions to the leaves. So this is how you would score total of 4 marks. If I go beyond, so they say is that there is an experiment in a plant that is tail crest plant. The species is Arabidopsis and if we come to this part B, they say that there is this experiment and they carried it in the tail crest plant. The genus is Arabidopsis and the species is Thaliana, and they were grown in the normal atmospheric and high carbon dioxide concentration. Further, they have to be telling us is that the transpiration rate, the mean number of chloroplasts per cell, and the concentration of the starch and the magnesium ions were measured. I have this tail crest plant. Okay. I am about to grow them in the normal atmospheric and in the high carbon dioxide concentration environment and the impact that it will have on their transpiration rate. the mean number of the chloroplasts per cell, the concentration of the starch, and the concentration of the magnesium ion. They measured it, so all of them, they were their dependent variable. Now they have given you the fill in the blank kind of a question for the total of 7 marks, and they say is that you are required to complete the sentences about the data which is shown in table 2.1. Now we have to be relating these blanks with the table. They say table 2.1 shows that increasing carbon dioxide concentration caused more starch to be produced in the leaves. Now here is a normal carbon dioxide, here is a high carbon dioxide, and here we have got the concentration of the starch. And what we see is that high carbon dioxide concentration, the amount of starch produces more, and in the normal this is less. This shows that at normal carbon dioxide concentration, the carbon dioxide we know since it is less here in normal amount, more here in excess amount. So in this it would act as a limiting factor for photosynthesis. They say during photosynthesis, dash molecules of the carbon dioxide are required to make one molecule of glucose, and we know from the equation of the photosynthesis that we require six molecules of the carbon dioxide gas to be making one molecule of glucose. Moving onward, they ask is that the greater quantity of starch stored in the leaves grown in the high carbon dioxide concentration means when needed, more sucrose can be produced for transport in phloem. So the leaves act we know like when we are producing more of the sucrose for the transport. So the sucrose would be carried from the leaves to the other part of the plant. So that part of the plant from where the sucrose would be carried away, that would be a source, and to what they are carried to, that would be a sink. They say greater the number of the chloroplasts per cell in the leaves grown in higher carbon dioxide concentration means that more energy can be absorbed from where we know as from the sunlight, and that sunlight as we know as that it would be transferred to the chemical energy. Further they say is that the transpiration rate is lower when the carbon dioxide concentration is higher. So here we have the transpiration rate. So when there is high carbon dioxide concentration, so the loss of the water by the transpiration is less. So that would mean that since the carbon dioxide concentration is high, so that would close their stomatas, right? And the loss of water by transpiration would reduce. So if the transpiration rate is lower, that would mean that the rate of loss of the water vapors from the leaf, it would be reduced. Further, they say that magnesium ion concentration is lower in the leaves because they have used the magnesium ions to make what? This is a fact that magnesium ion is related to making of the chlorophyll. Moving onward to this May-June 2024 question, this variant paper variant was 4.2, and this was question number 4 with us. And they say that some algae are single-celled organisms that can photosynthesize, and that would be right because the algae they are photosynthesizing as their cells they have the chlorophyll pigment in them. Now for 1 mark again they say is state the name of one mineral line that is needed to make the chlorophyll, and we know this is magnesium ion. If we just go to this B part, here are some of the students and they are investigating the effect of the light on the photosynthesis in the algae. And for that purpose, they are also using the hydrogen carbonate indicator. Since we have to see the impact of the light intensity on photosynthesis, so this would be our variable that we are required to be changing, so it is independent variable. Now they tell us themselves that carbon dioxide is an acidic gas, and if this is acidic gas, if its concentration would rise, so it would make the surrounding solution acidic, lowering its pH. They tell you is that figure this table 4.1, it shows the color of hydrogen carbonate indicator solution at different pH values. Now, if the pH of the solution is 8.4, the color of the hydrogen carbonate indicator is red. And if the pH becomes alkaline, that is 9.2, so here the color of this is becoming purple And here what we know is that the plants, they would photosynthesize. They would use up the carbon dioxide, and when the carbon dioxide would be used up, so the acidity would be lowered and the pH value would get higher. Since CO2 is acidic, so if I would remove it up, so ultimately the pH would increase and it would go more toward the alkaline side. And here you see for the pH 7.6, the color of the indicator is yellow. And now you see that 7.6 is the lower pH than the other two. And this is more like less alkaline, more CO2 would be available in the surrounding solution, or this would be released out of the plants into the surrounding solution. So that is why the pH is lowering. Now they say is that there are two test tubes and they were set up at the same time. Both had the algae and the hydrogen carbonate indicator. And when the investigation was started, so the color of the indicator was red in both test tubes. One of the test tubes, it was placed in the dark, and the other test tube, it was placed in the light. They say after 20 minutes, the content of the test tube in the dark were yellow, and the content of the test tube in the light were purple. What we know is that when the algae would be placed in dark, so they would not photosynthesize, they would respire. And when they would respire, so they would release the carbon dioxide into the surrounding hydrogen carbonate solution. And since CO2 is acidic, so it would reduce or decrease the value of the pH. However, when the algae would be placed in the light, they would photosynthesize and they would use up the acidic carbon dioxide from the hydrogen carbonate solution. So the CO2 value in the hydrogen carbonate solution, it would become less, and that would increase the value of the pH. And that is why in the light it's purple, the pH value is higher at 9.2 because the CO2 got used up. For the dark, this is yellow with the pH of 7.6, indicating that in dark the algae, they respired the CO2 into the surrounding solution. So when the CO2 would go out of the algae into the surrounding solution, and hence this is an acidic gas, so it would lower the pH value. For the 4 marks, they want you to explain these results. I have said so. Let us just look how you are required to be writing it. If some question appears like this in your final exam in the upcoming year, so we would say that for the test tube which was placed in light, the algae they carried out photosynthesis. And for that they used up the carbon dioxide which was available from the hydrogen carbonate indicator. And this would make the pH either you would say alkaline or less acidic. And that is why the color is becoming purple. Then I have written that for the test tube which was placed in the dark, so the algae they only respired, they did not photosynthesize. So the CO2 was released into the hydrogen carbonate indicator and it reduced the value of the pH or decreased the value of the pH. Why? Because the presence of the CO2 that is being released out of the algae, this is making hydrogen carbonate indicator more acidic. So that is why it formed the yellow color. Moving onward to the second part, they say figure 4.1 is a graph showing the effect of the light intensity on the rate of photosynthesis. So the light intensity is our independent variable and this happens to come on toward the x-axis. So anything that comes on the x-axis, at this point of the graph, this would act as a limiting. At this point, this thing labeled on the x-axis, it would become excess, and the other two external factors, which is the temperature and the carbon dioxide, so they would become limiting in this part of the graph. So for 3 marks, they are asking us that state which factors could be limiting the rate of photosynthesis at x and y. So here in this part, light intensity is limiting. And in this part of the curve, you see we have got a greater value of the light intensity. So in Y, the temperature and the CO2 concentration, they become limiting. For 2 marks, they have to be telling us is that starch and sucrose are made by the plants after photosynthesis. What we know is that the plant by photosynthesis would make glucose. If I would combine it with the fructose, we can make the starch. And if many of the glucose monomers, if they are polymerized together, we would end up making this complex carbohydrate which is starch with us. They say state the name of the tissues that transport sucrose around the plant and the name of one other biological molecule that is transported in this structure. So we know that the sucrose movement uh, from the leaves and all around the plant or in various parts of the plant, this is translocation, and this is done by the phloem tissue. Along with the sucrose, the other thing that can be moved by the translocation process. So this is amino acid. Moving onward to this question from the leaf structure. This happened to appear in the October-November of 2024. The theory paper variant was 4.1, and this was question number 1, A part with you. And they say is that this figure 1.1 is a photomicrograph of a cross section of a part of leaf. What I see is in this photomicrograph that this K that we are given here, so these are the cells of the upper epidermis. The N has got large cylindrical cells, so here we have the palisade mesophyll cells. In them we have got some rough shaped cell having multiple air spaces, so here we have the spongy mesophyll cells. For the cell, this would be the lower epidermis. The J could be the cuticle that lies on the outside of the upper epidermis cells, but we would see what the table has to say. And for 3 marks, they say state the letters in figure 1.1 that identifies where the gas exchange is controlled. So we know this is L because in the lower epidermis we have the guard cells having stomata in between them, contributing their role in the gaseous exchange. They ask is that most photosynthesis happen. So this is N because in the palisade mesophyll cells, the number of the chloroplasts kept is highest. And they say spongy mesophyll cells are found. So this is M with us because we see air spaces. We also see the vascular bundle here. And at the same time, these cells here, they appear to look rough shaped. So that is an indication of the presence of the spongy mesophyll cells. Moving onward to this question from May June of 2025, the variant was 4.3, and for your paper, this was question number 4A part, and this was first, second, and third part. They tell you is that this is 4.1, and this is a photograph of a water lily, which is Nymphaea alba, and that is living in a lake. Now, if we just go beyond, so they show you the photo micrograph, and that shows you the cross section of the water lily leaf. What we know is that here we have the upper epidermis. The B that they have given you, so this includes the palisade mesophyll cells. The unusual thing about is that in the land plants, we do not have air spaces in the palisade mesophyll cells. In the D, we have the spongy mesophyll cells, and they have got very enormous kind of the air spaces. They ask you for 2 markers that identify the tissue B and tissue C labeled in figure 4.2. So if you revert to the B, so these are the palisade mesophyll cells. And the C, these are the lower epidermis with us. Now, for 2 marks, they say describe two functions in water lilies of the area labeled D. So you see that the area labeled D, these are the air spaces, and the role of the air spaces is for sure in term of the exchange of gases, the CO2 going into these cells and the O2 releasing out into the air spaces, and that is equivalent to the gaseous exchange. So we would say that this carbon dioxide, so we would say that this T, the air spaces that we have, they allow the carbon dioxide to diffuse into the mesophyll cells for photosynthesis. The other thing is that these air spaces, they provide buoyancy, or they allow the water lilies to float onto the surface of the water so they can absorb sunlight. And for the last part, for the total of 2 marks, they say a green pigment is found in tissue B, and the tissue B, it was palisade mesophyll cells. And the photosynthetic pigment that we have with them is chlorophyll. So they ask us that you are required to describe the function of the pigment. So we know that the chlorophyll, it traps a light energy and converts it to the chemical energy that would make the glucose, or you could say it can make the carbohydrates for the plant. So I hope after such an extensive looking into the past paper questions from the plant nutrition, your approach of this chapter would be fine now, and you would be in the position to crack any of the questions in your upcoming exam out of the context that I have provided you in this video.